All right, good morning, everyone. I think we'll get started. We have a pretty packed schedule, so I want to make sure we get started on time. I'm Mitch Drum. I'm a faculty member here at Case Western Reserve in Genetics and Genome Sciences, and I'm going to be your host this morning. Before we do anything else, you've noticed the folks along the wall here and outside. These are our sponsors of the event, and I want to thank them so very much for their generosity, because without them, we would not have this. And I hope you got the chance to talk to them a little before we got started. After we're done, we have lunch. You'll have more chance to interact with them. So please help me make them feel welcome. More of the sponsors here. A special shout out to K&D Group. So they had the terminal tower lit up for us. So if you didn't notice, so thank you to them. So what we want to do today is just give you a sense of what's happening now in rare disease research, clinical care, all kinds of different aspects of what's going on. But I have to announce a couple special things from the host institution here, Case Western Reserve. This year we are celebrating 200 years. The Western Reserve College was established in 1826 out in Hudson, and then moved later on to the current location. And the Case Institute for Technology, or then called Case School of Applied Sciences, was started a few years later. 1967, they merged and became Case Western Reserve University. That big mouthful, but that's for your Trivial Pursuit game. The other thing is, some of you may have seen that Time Magazine announced what they consider, from their criteria, the top universities across the globe, and Case came in at number 25. And I think that's quite of an accomplishment, and we're very proud of that as well. All right, but why are we really here today? So this is to talk about where things are today. And as a geneticist, I'm a little bit more aware of what's going on in the genetic disease space. But there are advances not only in genetic therapies, but also in pharmacotherapy, so new drugs. And again, you can pick on these guys over here on the wall to get more details about any of the individual diseases. But I think I can say with a lot of confidence that we're now in an age of genetic therapies being a reality, so that we've now gone from can we do it to when will we get it done. And I'm pretty sure Dr. Anderson and some of the other speakers are going to touch on that as well. But another area we don't think about a lot is improved diagnostics. So it wasn't too long ago, if someone were affected by a genetic disease, there would be an awful lot of misdiagnoses because they're so rare a doctor doesn't recognize them. Now the ability to go in and sequence genomes changes that dramatically, and that's just going to improve as we move along. And new methods in drug discovery. You don't look at one chemical at a time now. You look at hundreds of thousands of them to see if you can find something that will work better. So things are really moving forward in a very good way. And I saw this article from NPR on Monday that the FDA now, because there are successes, things like spinal muscular atrophy, sickle cell anemia, and other disorders, that the FDA says the bar's now coming down a little lower, that we can get things approved faster now because we have successes and we have a sense of the deleterious effects, but also the potential benefits. So I think that's really good news as we're trying to move forward with these. And one of the other important messages I'd like to get across is you all are part of something that doesn't happen enough, in my opinion, and that is interaction between the clinical and research institutions right here in Cleveland. Here you have all four of us working together, and I think that's a wonderful precedent. So thank you for the folks from Case Western, from University Hospitals and Harrington Discovery Institute, Metro, and Cleveland Clinic. So thank you all very much. All right, and again, thank you to our sponsors. Again, it's just without you guys, this just wouldn't happen. We have a couple of proclamations. So this is being recognized at the city and the state level. So we have a proclamation from Justin Bibb, the mayor of Cleveland, who is officially proclaiming today as Rare Disease Day. Well, it's actually the 28th and 29th, if there were one. And then also from Governor Mike DeWine. So it's being recognized throughout. All right, so what are you going to hear today? Well, you're going to hear first from Tiffany Salmons about NORD, the National Organization for Rare Diseases. If you're not familiar with them already, you need to be. You're going to hear about genetic counseling. In my humble opinion, that's an emerging field that's getting more and more important every year. It's been around a long time, but what they can do nowadays is much more. You're going to hear about some laboratory disease or disease projects from laboratories, how we're advancing therapeutics for rare diseases. Justin Betamarco is going to talk about biosimilars, and he'll tell you what that word means. And I'm not sure, we may have been impeded by some travel issues, but if they arrive, you're going to hear about the use of artificial intelligence to help in diagnostics for rare diseases. And we'll have a panel discussion with patients, caregivers to talk about Tracy Greenberg will talk about patient advocacy. And then we have a special presentation during lunch by a genetic counselor who will be talking about neurofibromatosis. So, and then we'll get to network more. So without further ado, let me introduce Tiffany Salmons. So Tiffany gotten to meet several years ago through her work at NORD, and she is uniquely suited to tell you more about the organization. Okay, Tiffany.
Thanks, Mitch, and thank you to Pam as well for putting on such a wonderful and well-attended event, and for all of you for joining us today. So today I'm going to talk about NORD's Patients Assistance Programs because I feel like many in this room could probably benefit from them. And it's our largest team at NORD, and many people don't know what all we offer. So we have a dedicated patient services team in 3 different areas: patient assistance, information and resource services, clinical trial, travel, and lodging support, and a communication center that you can see the hours listed there. I love this slide because I think it really shows a lot of impact. So NORD awarded more than $21 million across all of our patients assistance programs back into the patient and caregiver community. So as a nonprofit, we operate on a very slim budget because all of our money goes back into the population that we serve. Our patient assistance team is our largest team at NORD, like I said, and they have, I believe, right around 25 to 30 people, where many of our teams are 4 to 6. And so that group of people answered over 135,000 calls last year alone, and emails as well. So I just think that call volume is a little bit insane and really speaks to all the people that we're able to help. And then 6,680 patients received financial assistance last year, so it benefited a lot of people. So our information and resource service team handles many community inquiries, including financial assistance, medication and medical expenses, health insurance premiums, genetic testing, travel and lodging, health insurance navigations through prior authorizations, denials, selecting the right plan, Medicaid unwinding, health insurance navigation, I just said that, repeating myself, rare disease specialists, rare disease centers of excellence, which we have many here in Cleveland, so I want to shout out to that. and mental health support and services, advocacy, disability, and immigration navigation as well. And sometimes what individuals need most is guidance, direction, resources, and a starting point. And so this is the team that really helps people in that time of need and try to figure out, where do we go next? If you're interested in trying to find out more about these different programs, there's an email and our phone number that you can reach this team at. As far as our comprehensive support, we do help with clinical trial support and assistance, expanded access, premium and copay assistance, medical assistance for out-of-pocket expenses not covered by insurance, medication assistance, travel and lodging assistance. We also have a rare caregiver respite program, which I'll talk about here in a little bit, and rare disease educational support to attend things like this. So this is our Rare Caregiver Respite Program, and I think it's one of my favorite things that NORD offers. I'm a caregiver myself, which I'll talk about here in just a second, but NORD offers this program for caregivers. You can use it in one large sum. You can spread out that amount awarded throughout the year. Whatever a caregiver needs to kind of rejuvenate themselves and get care that they feel safe with their child receiving, true respite providers. So then they can come back, kind of with a fresh mindset and ready to do this. We know caregiver burnout is a real thing, and so this is a really important program, and it is open right now. So if you're interested in finding more about that, please visit our website, and you can see that, that program and how to apply there. So as I said, I'm a caregiver. So my why is this little guy right here. His name is Rowan. He is 8.5, and he is a Spitfire little redhead. I'm not really sure where he gets that from. But I feel like many people aren't born into rare disease advocacy. There's usually some sort of spark that lights the fire inside of you to fight this fight. And for me, that was my son, Rowan. The day he was born, he came into this world as a 4-pound, 14.2 ounce, very floppy, as they called it, not hypotonia yet, baby boy. And it took 21 extremely long months of back-to-back testing to simply give that thing a name. That thing that was trying to define my son relentlessly. But as many of you know, there was no immediate manual or treatment guide or plan of what to do next once we found that diagnosis. We went through microarray testing, brain MRIs, countless panel tests, and EMG. Could it be SMA? Could it be Prader-Willi syndrome? What about CMS? How about a myopathy? That list went on and on and on until finally we reached the point of whole exome sequencing. And after 6 long months, 182 days, in case anybody else has counted that, that seemed like an eternity awaiting results while continuing with neuromuscular testing throughout that duration, we finally found our one-fits-all holy grail diagnosis, Coffin-Siris syndrome, a multisystemic connective tissue disorder. However, our geneticist's words to follow haunted me for quite some time. It is very rare, and we don't know much about it at all. There are only 144 registered cases in the U.S. as of now. Though even still, that was a huge weight lifted off of my shoulders, just knowing all of the things that it wasn't. At least now we had a name to define this disorder that was trying to define him and his future. However, that and a 5-page list of encompassed symptoms is about all we did have. No cure, no treatment, no manual. Just continue to treat symptomatically is what she told us, along with adding a few more specialists to our already lengthy list. He now has 26 specialists. He has a village that I never really dreamed of being our village. It looks very different. But what it did give me, that diagnosis, was NORD, which gave me a community. And as a parent of a child with complex medical needs, Google becomes your best friend and your worst enemy. And despite how many times each doctor specifically told me, now whatever you do, do not go home and Google. Of course, that's exactly what I did because they didn't have much information. But what it did provide when I did Google was NORD's rare disease database, which I think is one of our most excellent resources we have to offer. And so if you haven't visited that, please take a chance to visit it. I now have the pleasure of getting to add new rare diseases to that database through my work at NORD. And that gave me the best and most accurate description of Rowan's diagnosis I've found to date and continues to get updated every 2 years. It also gave me the opportunity to meet others who had gone before me, who had knowledge and experience I didn't yet possess through events just like this. This community is so powerful, driven, and dedicated to changing outcomes, and I want to sincerely thank each and one of you for participating in this event with us today and for being a part of this incredible community that is quite large despite being so rare. NORD's motto, "Alone we are rare, together we are strong," and this event reminds me each year that it takes all of us working together to move the needle forward: clinicians, researchers, advocates, patients, caregivers, academia, industry, etc. So I hope you all leave here inspired by the stories and advances that you hear about today, and remember this event and this moment as a pivotal moment in your journey and as the spark that inspired you to help move the needle forward for the rare disease community.
All right. Thank you very much, Tiffany. Before we go to our next presentation, I have to go over the things that I forgot about. And one of them is for those of you who parked in underneath the building here, we still have a few QR codes that can get you out of jail for free. So we can get those at the front table. I also need to welcome our online participants. So we're being live streamed today. So thank you all for joining us that way. And then, as Tiffany had mentioned, Pam Judge needs to be specifically called out because Pam is the driving force every year for this event. So thank you, Pam. All right, so our next presentation is from Becky Dara. Becky's a longtime colleague of mine at Case. Becky is not just a genetic counselor, and co-director of the counseling program, but also a molecular biologist, geneticist, and she graciously agreed to bring you up to speed on what genetic counseling is all about today. Okay, Becky?
You forgot one announcement, though. He forgot one announcement. Today is Dr. Drum's birthday. Happy birthday! He does not look happy that I said that.
Thank you for coming to my party.
You're right. So, all right. I've done away with my ability to move the slides here, haven't I? Well, who is my brother? There we go. All right. Okay, so I will start by saying, if you're in the room and you're a genetic counselor, a genetic counseling student, or a genetic counseling assistant, please raise your hand. Oh my goodness, so woohoo! Most of the people in this room know everything I'm about to say. And if you have further questions about genetic counseling, please find, hands up, one more time? Okay, find any of these people with their hands raised, and they can elaborate more, I'm sure, on anything I'm about to say. So I know we have a number of people online as well. Take a peek. There's a lot of genetic counselors in the Cleveland area who can help address anything we're about to talk about. So this is a definition of genetic counseling, and if you're into reading the fine print, the citations that I force all of my students to include on the slides, you'll notice it's from 2006, which seems like a really short time ago to me, but it's actually 20 years ago, right? So I chose this definition somewhat intentionally. This is from Bob Resta, and it was with a working group with the National Society of Genetic Counselors, put together this definition. I think it's a very good definition, but we're going to talk about one thing that it's missing. So in this definition, we talk about genetic counseling as the process of helping people understand and adapt to the medical, psychological, and familial implications of the genetic contributions to disease. And within that process, we think about interpretation of family and medical histories to assess chance for disease occurrence or recurrence. We talk about education, about inheritance. How is this moving through families? What is the risk that you might have another child or that your child might have a child with something similar? Testing, which is a big thing we're going to talk about quite a bit today. Management, prevention, resources and research, and then counseling to promote some sort of informed choices, adaptation, risks, adaptation to that risk, how we're going to live with this moving forward. What can we do to make sense of this information and to integrate it into our lives. What we don't see on here, anybody know? What do you think might be missing from this definition? Treatment, right? So in 2006, there really weren't. So that's not just an oversight in this definition. There really weren't a lot of avenues to treat genetic diseases. And that's changing, and that's super exciting. I will say I'm very excited and proud that last year, for the first time, we in the genetic counseling training program developed and started a therapeutics course. There was enough success in this space to necessitate an entire course on therapies for rare genetic diseases, which is, I think, super exciting. We're going to give a little clap for that, because that's the work of many people in this room and people on the sidelines over there who have sponsored this this talk as well. So I think we'll just keep that in the back of our head, that there is success and there is motivation and progress, and all of that is happening in terms of treatment of rare diseases. We heard a few moments ago a very wonderful example of what we refer to oftentimes in the genetic space as the diagnostic odyssey. And I just want to highlight that, this is a big deal. So Tiffany talked about an 6-month diagnostic odyssey for her family. But on average, families wait about 5 years, if you can imagine, to get their diagnosis. And that can really take its toll. And that is definitely a place that genetic counselors can help, and I'll explain a little bit how. That diagnostic odyssey, let's talk a little bit about that toll, because I found these figures a little bit staggering. So on average, about half parents say their friendships declined. They report feeling socially isolated. They felt anxious, fearful, overwhelmed. They're receiving treatment, oftentimes for anxiety or depression, and almost half say they aren't coping financially with that cost of care. So these are enormous burdens for any family, any individual or family to undertake when going through this process. And this is something that we deal with quite a bit as genetic counselors in trying to help our patients and our families navigate. So what can help with that process? What can help with diagnostic odysseys and getting to the place where you have that answer that may be important? There are 3 general areas: education, streamlining the process, and a push for better policies. And genetic counseling sits at the forefront, I think, of all of those. We've already talked about the education component for genetic counseling. Families and providers need to know that testing is an option and that it can help. Many professional societies recommend exome or genome testing as a first step, maybe as soon as symptoms appear, and that might be able to speed up the process a little bit. Testing and counseling and results should feel connected. And I think that's a big one, and that's a big space where genetic counseling comes into play. I think sometimes what makes some of these diagnostic odysseys even tougher is this doctor has ordered this batch of tests, and then this one has ordered this, and there's nobody really connecting or organizing, making the whole process integrated and thinking about the results as they relate to each other, not just as individual systems being tested. And then pushing for better policies. So genetic counselors do exist in the advocacy space, as does NORD. So in a way to prevent insurance delays, those can cost families time and care, and advocate for earlier access, broader coverage, and more equitable testing, which is a huge deal right now. So genetic counselors help patients and families navigate those testing options, make sense of the complex data, and provide emotional support. Crucially, connect families with others dealing with the same condition. So even if there are only 140 children with a similar diagnosis in the US, genetic counselors can help find those families. And there is a lot of value in just meeting somebody else who's going through something similar to what you're dealing with. I've had many people come back and say that was the most impactful thing, was getting to talk to another mom who I didn't have to explain all this to, who knew exactly what I was challenged with at that moment. So that's a big space that we can help with as well. There are some myths of genetic counseling that I want to go ahead and dispel for any of you who might still be thinking about those myths or who aren't familiar with that. The myths are here on the left, and then we'll address each truth as they come up. The first one is that genetic counseling is only for pregnant people. And you will have no idea how many times we hear that, that's really for when you're trying to decide about a pregnancy or you're trying to figure out a risk for a pregnancy. pregnancy and prenatal genetic counseling was maybe what the field was initially born out of, but it's so much more now. Genetic counseling is for individuals at any stage of life, including before you're pregnant, preconception, pediatrics, adult onset, and the cancer space. Genetic counselors see patients in all of these areas. As I already mentioned, there are also many genetic counselors that are involved in the lab and testing side of things, and also in advocacy, policymaking, education, teaching other genetic counselors. The list goes on and on for genetic counseling roles. Another one we hear often is genetic counseling or testing will make me uninsurable. That's a common myth, a common concern. And of course it's a big concern. We've already talked about the financial burden of a lot of this, and thinking about that burden in a way that makes sense and trying to mitigate it as much as possible is a huge goal. The Genetic Counseling or Genetic Information Non-Discrimination Act, also known as GINA, will help with this. It does protect against discrimination by employers and health insurance companies based on genetic test results. I will say that the same is not true for health insurance or long-term disability insurance. So that is something we discuss with families before they have testing oftentimes is it worthwhile to secure some of these, especially for adult onset things. We might talk about securing insurance prior to testing. But in terms of health insurance, no, there are protections for that. It is commonly thought that genetic counseling and genetic testing is expensive. That is not true. Genetic counseling and genetic testing is more accessible than ever. Most insurers cover it when medically necessary. Genetic counseling in most states now has the ability to bill for their services. Insurance covers those services. This is not a just for the elite out-of-pocket kind of situation. In some cases, cash pay options are available for around $250 for a lot of genetic tests. The out-of-pocket costs often have caps. So even if your insurance denies it, the max you will pay is some amount that you would be able to know ahead of time. This is a big one, that at-home tests are just as good. I did that 23andMe and I got results, so I know that this isn't genetic, or whatever's going on in my family isn't genetic. Oh, sister, that is, those tests are not comprehensive. They do not provide full risk assessments, and they cannot replace the expertise of a genetic counselor for interpretation or a clinical geneticist. They are shiny and new and cheap, and all you have to do is spit in a little cup and send it off in the mail. And sometimes there's a Groupon, for this. So this is an exciting avenue for a lot of people, and a lot of people, I think, got mistakenly led into thinking that was the same kind of genetic test that would be done clinically, and it's not. So those are not just as good. Maybe if you want to know if you can smell asparagus or whatever, it might be as good. But for an actual clinical result, no, don't take that one. And then I have no need to see a genetic counselor if my doctor already did all the testing. That's a misconception as well. Genetic science, as we know in this room, right, we'll hear about today, advances extremely rapidly. There are always new reasons to consider retesting, a different testing approach. Consulting with a counselor later may be necessary for this updated information. Even if you had results that came back, there are these ambiguous results called variants of unknown or uncertain significance. Those are always being reclassified. It's always worth a trip back to your genetic counselor, maybe a year, maybe 2 years later, to say, what's new, what can we do if I didn't get the answer that I needed to take care of the care for my family. So those are some common myths. Hopefully I've convinced you that they are in fact myths, and that we don't need to be as concerned about those for genetic testing or genetic counseling. But if there are others that I haven't addressed, please find a genetic counselor in the room and ask them what their thoughts are and how they can help. So besides in this room, where can I find a genetic counselor? There are genetic counselors at most major hospital systems, and certainly all the ones in Cleveland. So University Hospitals has a whole team of genetic counselors. I should have paid attention to the numbers. If anybody knows how many genetic counselors are currently employed at UH, does anyone have that number? Nobody in this room knows. Maybe 10? Okay. And the Cleveland Clinic? 35. Okay. And Metro Health is 6? Okay. And do you know, Monica, on Akron, how many genetic counselors are at Akron?
It's similar, maybe around 10.
About 10? Okay. That is a wealth of genetic counselors just in the Northeast Ohio area, especially for a city the size of Cleveland. Our cup runneth over, definitely, on genetic counseling and genetic access to care. So there's no excuse, people. Go find your genetic counselor and get the care that you deserve. All right. Thank you.
Don't leave just yet. So I'm a bit obsessive about staying on time, but we are a few minutes ahead, so we do have time for a question or two for either Tiffany or Becky, if anybody has them.
You meant that.
I did, yeah. So, no, we got a shy audience today. Oh, no, sorry, Kumar? And I'm going to repeat it, so go ahead.
I was wondering if you can comment on people using chatbots, or using Google Gemini to put information in, and so just a comment on that.
Yeah. So the question is, can you comment on the use of chatbots and such things?
Yeah.
Yes, you can.
I can, yeah, right. I'll give a chatbot answer. We went through this when Google was invented, right? And now it's when chatbots are invented. I think there is a space to use that information, but we are actively teaching our students, and I think much probably to their frustration, including a lot of assignments where they have to go to ChatGPT, ask the question, and then vet the answer and come up with that. So we do. At least I talk with my patients about that. What have you found? What have you heard? What was the source? Can you tell? Was it through chatbot? There is good information. There are ways to use it responsibly and that can provide decent. It is just, I think it's almost impossible for the everyday person to know what's real and what isn't. And so that is a way that your genetic counselor can help. Yeah.
We've heard a couple examples of exome sequencing informing disease. Tiffany, thank you for sharing. But there's a large amount of non-coding region, and much of risk with these genome-wide association studies and others find it in these non-coding locations. Right. Is that working its way into the genetic counselor's diagnostic toolkit?
Can you repeat that?
Yes. So the question was, with the exome results, we have a lot of variants that show up in non-coding regions. Is that something that the genetic counselors are trained to interpret or to look at or to at least consider? I would like to ask my genetic counseling students, how much do I talk about the non-coding region? How many talks are titled, "It's not just the coding region"? Many of them. Yeah, so that's a great point. And I think we talk a lot about the difference between an exome versus a whole genome, what you're going to miss in an exome, why all of the disease-causing variants we know so far are in the exome, because that's the only place we've looked. So there's a lot of conversation about how we're going to interpret these regulatory regions and the non-coding regions. But I think that's going to be the next huge frontier. It's 98% of the genome is non-coding, and as you know, so thinking about that in a practical way is a daunting task. Yeah, please.
I'll just add that I've also moderated a panel on why genetic testing is important, even post-diagnosis. For example, my son just gained a new rare diagnosis 3 days ago as an incidental finding. So with that, I will say that one of the things the geneticist on that panel said is the more we know about genetics, the less we know. But I think that things are advancing so quickly that having that information on the forefront will help inform the future. So just knowing those things, even if we don't have all the answers now, it's helpful for the future.
Absolutely.
Any others? Measure?
In terms of, we have so much, why aren't all of these things in a regular panel where, someone who's pregnant can get tested? What's holding that part back?
Yeah. It could be cost, but we have to remember that part of this is choice, and not everybody wants all this information. So there aren't and that's a big part of the genetic counseling process is what do you want to know? Not everybody, we're sort of, I think, as scientists, we are perhaps At least I know I'm a little bit biased. Well, of course, all the information is better. Why wouldn't people want to know? Actually, clinically, there are a fair number of people who don't. People who don't want to know the incidental findings, people who don't want that information. And so we have to remember that. So I think that is a big barrier to having. But having said that, there are more and more things that are becoming standard. But I think choice is still a very important part.
So I'm just going to add on, I think it's an important point to understand how fast technology is changing and what that's doing to the landscape. So the first genome draft, when it was produced, took 13 years, $3 billion. Now in 24, 48 hours, we can get a whole genome for a couple $100. So what that does to make things clinically possible is just huge. So anyway, so fasten your seatbelts. So, okay. All right, so our next speaker, Marissa Scavuso, another colleague from Case Western. Marissa, I'm not even going to call you a rising star. You're just a star in the intestinal disorders. And did I step on a cord?
Great, awesome. Well, thank you so much. I'm excited to be here and be able to share some of the work that my lab does. So we focus on the gut and how it functions in both health and disease. And specifically, our lab focuses on the gut's nervous system and a cell type there that's often overlooked called glial cells. I'm also excited to be here because I'm going to be sharing some of the work we're doing in neurodevelopmental disorders, some rare syndromic forms of autism. So both as a scientist and a mother of a child with a neurodevelopmental disorder, I'm really passionate about the work that I'll tell you about. So you probably don't think much about your gut when it's working, but history has shown us that when it's not working, that's pretty much all you could think about. So Napoleon was the commander of the French army. He was also chronically constipated. And the reason why in 1815 it's thought that they lost the Battle of Waterloo is because he couldn't get on his horse because he was in too much abdominal pain to sit comfortably. So the fate of Europe at that time didn't really hinge on how you're thinking, but really how your gut's functioning. So the next time you think that the gut's not that important, you can just remember that the health of the gut has already changed the course of European history. And while gut dysfunction is not at all rare, it's actually exceedingly common. Most rare diseases, especially neurodevelopmental and neurological conditions, have gut dysfunction. So this is actually something that's seen quite a lot. And when you look at the gut, so I know most of you probably don't study the gut. I'll walk you through this really slowly, and you don't need to know every cell type here, don't worry. But if you take a cross-section in this little diagram here, this is where food would pass through. If you're eating something, it's going through this area. You have all of these different cell types that are there. It's an incredibly diverse tissue. There's many, many different cell types, even different species that are there, that are all coming together to make the gut function properly. This also means that there's a lot of opportunity for things to go wrong and a lot of opportunity for dysfunction. And our lab specifically, like I mentioned, studies the gut's nervous system. And these nervous system cells are composed of both neurons and these often overlooked cell types called glia. And these are really everywhere in the gut. They're in every layer of the tissue from the outside in, from the beginning to the end. So again, when you think of opportunity for dysfunction, these cells are everywhere, controlling almost every function that the gut has, and can be really, disruptive if they're not working properly. And one thing that's really interesting about the gut's nervous system is actually it's really independent. So it doesn't need the brain to function. So if you take intestinal tissue out of the body, so this is a mouse's intestine that we took out, we stuck it in a dish, and this is 3 days later. We didn't add anything to here to make it do this. It actually just keeps doing this on its own. And that's because it has this nervous system embedded within it that can function without the brain. This is often called the brain inside your gut because its complexity and its ability to do these types of things, to keep things moving even without the brain. So we're really, really fascinated by how this works and how it doesn't work. And with this in mind, we wanted to ask, how can we really model such a complex tissue, this really cellularly diverse tissue in the lab to model rare human diseases and conditions, specifically thinking of neurodevelopmental and neurological conditions where we know that these gut disturbances are extremely common. And we wanted to do this in several different ways. One is maintain this complexity, so have a complex tissue that's three-dimensional, that has all these different cell types present, that's human, but without all of these other confounds, without the brain, without the microbiome, without all the weird stuff you put in your mouth and the delicious eggs we were eating earlier. So how can we do this in a reductionist form, but still keep that complexity to understand it? And importantly, for human disease and human disorders, how can we do this in a way that we maintain the genetic complexity of the donor? And one way we could do this is with human pluripotent stem cells. So many of you might already know what this is, but you could take stem cells or tissue from any individual. This could be blood cells. It could be a little skin punch. Actually, my friend during our PhD, we took a skin punch from her and made her own stem cells, and I actually have them now here, which is kind of fun. So, but anyways, we could take this from anyone, essentially, and reprogram these cells into a state where then they can become anything we want them to become, depending on the signals that we add, in theory at least. And luckily, in practice, we can do this in the lab. We can actually turn these human stem cells into three-dimensional multicellular gut tissues. And these are matching all of the things I just told you. They're complex. They have many different cell types present. There's no bacteria. There's no brain. But we're able to actually watch the tissue form and develop and start to work on its own in all of these different conditions, keeping in mind the genetic background of the donor. And here's just a video of some of these tissues that we've generated in the lab. So these are three-dimensional videos going up and down throughout them. You can see this hollow center. This is actually where your food would pass through. This is the lumen of the gut. And this red lining is the epithelial lining. So this is what lines the inside. It absorbs your food and digests everything. And importantly for us, we're able to do this with the nervous system. So we see both neurons and these cells. We're really interested in glial cells present throughout. So they innervate these miniature human gut tissues that we're able to grow in the lab. So in theory, I could take any of your cells and make your gut in the lab. So this has been really great. This allows us to then look at gut development at early stages, but especially if we're interested in these rare neurodevelopmental disorders, we need to understand more about how this is actually forming over time, so a more dynamic process. And one way we could do this is actually through surgically transplanting these into the mouse kidney. This allows these to mature more. So at early stages, these are more like human fetal tissues, human fetal gut, but then after it's more like the gut of an early child. So here you can see the differences. Here you see these nice red circles here. These are actually opposing muscle layers. These actually contract on their own in a dish, which is really weird. And you see importantly for us, this formation of these neurons and glia into these really organized web-like networks that form these robust connections. So now we can actually watch the nervous system in the gut form, make connections, and develop. So this is really, really important. It gives us access to something that otherwise is completely inaccessible. So to access human development in the gut in the context of these rare conditions. So this has really been an exciting system that we can apply to a lot of different things. And one of the things that we have been applying it to is autism spectrum disorder. So I do want to be clear, autism spectrum disorder is not at all rare. It's actually pretty common. I think now it's about 1 in 32 people in the United States might have autism. However, there are rare syndromic forms of autism. So these are forms of autism that can be attributed to specific genetic variants, so specific genes that have changed that increases your risk. But really importantly, these genes are not causative. So some wonderful work done here at Case Western by Tony Winshaw-Boris has shown that individuals with this PTEN mutation, if you correct this mutation and you grow from stem cells little brain tissues, it doesn't change everything. So the genetic background is still really important. So we really still need to do this from these complex stem cells that allows us to maintain this. So we're really interested in this, and especially because gut issues are extremely prevalent in autism, including these rare syndromic forms that I just mentioned. And when you think about it, in the brain, where we know these neurobehavioral symptoms are occurring in conditions like autism and in these syndromic forms, there are neurons and glia, and these neurons and glia are disrupted. But in the gut, we also have neurons and glia, and a lot of the same genes are expressed in these cells in the gut. So this actually provides a genetic opportunity for them to become dysfunctional on their own. So over 90% of the high-risk autism genes are disrupted and expressed in the enteric nervous system and this nervous system within the gut. And one of these I mentioned already, PTEN, is one of these syndromic forms that I'll talk mostly about today. So we took these stem cells and then we grew their three-dimensional guts in a dish, and we did this from this individual with this syndromic form of autism, as well as neurotypical controls. And I'm not going to go through the genetics, but just to say these dots here show that we mapped every single cell in these tissues as they developed and formed, and allowed us to look at what the cells were saying to us, to understand more what is actually changing between the tissue, the nervous system in autism and in these neurotypical mini guts. So we looked at this, and I'm just going to summarize the results that we found. We found that early in these early tissues, early in development, that actually the connections in neurons were decreased. So there were lots of signs that there were decreases in the connections that were occurring. But then we, unusually later, we didn't expect this. We actually saw the opposite. Suddenly there was way too many connections forming. So this was really interesting because a lot of the gut symptoms that we know are common in autism include things like defects in gut motility, constipation, diarrhea, and abdominal pain. If you have too many connections in the gut, this could actually be causing that. But then this made us wonder what could actually be driving this? What could actually be causing this to happen? Obviously, this seems like something that's inherent to the tissue because we're not adding anything else. We're not adding environmental toxins. We're not adding the brain. We're not adding bacteria. This is something that's actually programmed there. But we wanted to know what was actually going on. And one of the things that's really interesting, and I'm really proud to be part of the GLIA Institute here at Case Western, is that in the brain, we know that glia can change and do different things than they're supposed to. So normally glia are supposed to do things in the brain that help neurons function, but sometimes they can actually change in many different conditions and diseases, including in autism, and transition into either these neurotoxic types or these types that secrete different things to help promote neuron function. So we wanted to know, okay, we saw that neuron connectivity was changed, but could these glial cells actually change too and be doing something? So we took these gut tissues and we looked at their glial cells. And in green here, these are these, on the bottom, this is a gut tissue from this individual with this syndromic PTEN mutation. And on top, this is a neurotypical control. And you can see the green is much higher on the bottom. So this green is an indicator that there's more change occurring within these glial cells. And we checked this in independent experiments because we're really rigorous and thorough. And it kept showing up again and again and again, showing again that these cells were changing in the gut no matter what, like when we're growing them in the dish in the lab. And when we looked to see, using these maps that I showed you, what was actually happening, we were really surprised that it wasn't actually this toxic subtype that was emerging. This wasn't, the change wasn't that these cells were actually damaging neurons. It's actually that they were secreting a bunch of things and expressing a lot of things that actually should promote neurons to connect and form connections. So what we think is really happening throughout development in these syndromic forms of autism is that early we have a decrease in synaptic connection. So these neurons are not connecting like they should. So then these glial cells kind of freak out. They see this happening, somehow they sense it, and then they start overcompensating by secreting and releasing all of these different things that then cause later the opposite. So you have this really asynchronous development that occurs, and then it ends up having this dysfunction that's programmed within the gut. And we're actively testing out what this actually means for these individuals. Like what does this mean in terms of their gut's function? Does this actually translate to changes in how things form? So we're looking at how these cells are active in the dish, looking at their firing, and looking at how the gut contracts. And here you can see in the middle, this is actually a video of one of these in a mouse kidney capsule that's moving around on its own. So this allows us to look more at how the gut is changing in these rare syndromic forms of autism. So without the brain, without the microbiome, the tissue, the nervous system there is actually wired differently. And this is really interesting. But this is a rare syndromic form of autism. This is one individual, one donor line that I showed you. So we wanted to know, is this actually, since we know gut conditions are really common across these rare disorders, could this actually be something that's happening and conserved across these? And to do this, we took stem cells from this really wonderful resource, the Simons Foundation, which allowed us to get all of these different rare syndromic forms with these different risk genes that you could see here. And what was really cool about this is we can also track every single thing that these individuals have experienced in the clinic. So we know that these individuals have robust gut defects. And when we look, this is all very, very early, so you guys are some of the first people to see some of these results. But when we look to see if these glial cells were changing at this early stage in these other rare forms, we've already seen evidence of that. So here in gray, this is neurotypical organ mini guts. This is the level of expression of these factors that I mentioned change that are kind of like overcompensating. And what I already showed you is that this increases in the syndromic form where we have a mutation in the gene PTEN. But when we look in this other syndromic form, we see it's actually even higher. So this indicates that maybe this is something that's happening across all of these different rare subtypes. And by taking this type of information, we could actually try to use this to our advantage to understand what are the common mechanisms that are happening and the pathways that are driving this dysfunction across rare conditions. And this will allow us, by using many different genetically diverse donor cells, to kind of identify what's actually happening here and how we can actually therapeutically target this. So we don't think that these gut comorbidities are just a coincidence. It seems like it's because of many of these genes are expressed there in the nervous system in the gut. And as I mentioned, many rare neurological conditions have gut gut disturbances. And we've found that there's evidence that these glial cells that are often not even looked at are changing, and that this seems to cause that these neurons are not connecting correctly, and it's causing this change in development. We're currently teasing this apart, trying to understand how this is happening and what's going on with this, what's the consequence to individuals. But overall, using this type of system and having access to this type of approach where we can grow tissues from individuals with these rare diseases and rare conditions, we can do things like try to understand how it's happening, develop new therapies, and potentially actually alleviate some of these pains that they experience. And I want to close by just quickly mentioning, I know this is a broad audience, but it's been a tough year to be a scientist because a lot of the things that are happening around us, but having events like this is just so important. You know, doing things like doing great science, which we try to do every single day in the Skavuza lab, but also sharing that work with the community, sharing it with each other, and talking about these things is really important. One other thing that I'm really passionate about is investing in the future of science. And just really briefly, wanted to mention that there's a nonprofit here in Cleveland that was co-founded in 2019 that really focused on low-income public schools to try to increase the opportunities in science and the participation in science by different people. And we have major programs. One of them is called Life as a Scientist program, where we actually have over 100 volunteers from Cleveland Clinic, Case Western, Cleveland State, industry, pharma, that come into the classrooms every week and work with these students to allow them to actually do thesis projects themselves. And this is really exciting because then they get to ask questions and do all of the things that made me fall in love with science. And at the end, they publish their work. We also have paid internships that happen, and we take some of these students and have them come work in research labs over the summer for several weeks, which has been really, really rewarding. So if you're interested in this type of stuff, we're always taking volunteers, and we're always interested in having more people do this type of work with us. But with that, I just want to thank everybody who helped do the work, all of our funders, and Mitch for the invitation to share this.
What is that to know if the same patients you've looked at in India, in gut, do they also have similar effects in terms of you see more synapses or connections in the brain for these individuals, if anyone knows if that's one of the phenotypes?
Yeah, that's a great question. I'll repeat it for those online. So Mayur was asking whether or not some of the phenotypes we saw with changes in glial cells and also neuron connectivity was also seen in the same lines of people have looked in brain tissue. So actually right here at Case Western, we're really lucky to have Tony Winshaw-Boris, who has done a lot of this work, and we actually got these stem cells from his lab, these P10 syndromic lines. So what they did is they took them and they grew miniature brain tissues in the lab, which allowed them to look at how the brain is developing in this form of autism. And what they found is really similar, that there were defects in synapse formations. I don't believe that they looked into glial changes, but they do have the single cell data, like those maps that I showed. So that's something that we are actually actively looking into, is grabbing some of their data and comparing to what we see, because if it is a conserved process in both tissues, then if we find something that can help the gut, it might be able to help some of the neurobehavioral symptoms as well. Yes.
Really exciting.
Thank you.
Very inspiring. I was wondering if, sporadic, most autism is sporadic. In fact, we don't have a clear cause. Maybe it's hidden in that non-coding region. But, one question is your, intriguing change in the glia transcriptionally. If you look at the sporadic autism, there's large data sets on, sequence changes. Are you seeing any similar signature?
That's a great question. So the question was, most forms of autism are idiopathic or sporadic, and we don't have risk genes that we know are actually potentially driving or increasing the risk for developing these neurobehavioral symptoms. And whether or not in these sporadic forms we're seeing some of the same phenotypes, right? Some of the same molecular changes. So we haven't looked in idiopathic forms. We do have access to those types of cells from the Simons Foundation. We've kind of tried to first do a panel of more genetically defined ones, but then we would love to expand to that. I think the more we could, although my students are going to hate me for continuing to say we should do more and more, but the more genetically diverse lines, and even those that don't have specific genetic variants that we know of, might be in the non-coding region, we don't know, but the more we can add into here, the more I think we could really funnel this down to really understand and identify these central mechanisms that might be conserved across them. Because I really do believe probably, and it's not the same risk genes, of course, that are causing things, but it's probably similar pathways that are happening and similar processes that we can think about. Yes?
With P10, we know that the genotype phenotype is PTEN, but then there can be different phenotypes like Cowden syndrome
Mm-hmm.
and Proteus. Like, is this research mainly with people who have the symptom, like the clinical presentation of autism, or is it with others?
That's a really great question. So the question was about PTEN is it's involved in a lot of different types of syndrome. So one of them I mentioned was PTHS. You mentioned Cowden syndrome. So whether or not we're looking at other forms of PTEN mutations and what is changing, or just looking at autism spectrum, we're mostly interested in autism spectrum, which of course that's an umbrella term. That's just based on behavioral symptoms. It's not really like it's a condition, but it's not like defined. I think looking in these other things would be really interesting in PTHS, and that's something I'd love to do in the future, but just not a current focus of the lab. But that's a really great question. Yes.
I have a quick question for you. I believe you mentioned in your autism model for increased synapse formation. I was curious what your thoughts are on that. Is it not enough synaptic pruning, dysregulated pruning, anything?
That's a great question. Did they hear it online with the mic? Or should I repeat it? Okay, great. Okay, so I won't repeat it. So there is pruning that occurs in the gut. So macrophages in the brain, microglia prune synapses during postnatal development in the gut. Macrophages do this. That's really, there's only been one study that shows it, and it's beautiful, I believe it, but all very emerging. We don't have macrophages in our culture system. So that's one thing that is limiting in this. You know, it's very reductionist, which allows us to look at this, but it could be that maybe some of these synaptic connections would be somewhat mitigated if we had those, or maybe not, maybe it would actually be worse. What I think is happening is that these glial cells somehow are sensing that early there's not enough connections forming, and then this reactive phenotype that they have, they're secreting a bunch of things that actually cause overcompensation, and then suddenly you have way too many connections. So it's kind of weird because early we were super confused when we saw this completely shifting opposite phenotype, but we're trying to do some experiments now to like change glial cells and see if this can actually drive it. Doing things like chimeras, where we take like glial cells from an individual with autism and put them with neurotypical neurons and see if that actually forms too many connections, if they actually are going to cause this. So lots of work still to be done, but that's a really great question. Thank you.
All right. I am happy to introduce our next speaker, Dr. Matthew Anderson. Dr. Anderson and I have known each other 30-some years now. I got to know him when he was an MD PhD student at Iowa. We were both working in cystic fibrosis at the time, and I think gives us a wonderful perspective on where things are going, where we are now, and what the future is going to hold. So Matt, please.
Thank you, Mitch. Yeah, I was about to say the same, actually. It's an interesting kind of circling back and in the right setting too, you know. So I have had a little bit of a circuitous career going from cystic fibrosis really in my formative years in research, which was really an honor, honestly, to deciding to go to the nervous system, which I felt was more complex. You saw a sense of that just now. And then the richness of genetics in the nervous system has really just blossomed. But then I decided I wanted to do drug development. So after 20-some years in Boston, running my own independent laboratory, I went to Regeneron to lead the neuroscience program there. Learned tremendous amount about their approach to these genetic disorders. You know, what are the new next wave cutting edge approach? But I did learn an important fact, which is that they can only take on so much rare disease because of other obligations that they have. But they're using rare disease as a way to get insight into what the right therapeutics are for common disease. Just like we heard, autism has a subset explained readily by genetics, but there's a lot that is not. Well, the same is true for Alzheimer's. There's early onset Alzheimer's that has a clear genetic thing. And that was really the approach that they were taking towards Alzheimer's, Parkinson's, other things. So let me get into what I'm now doing at the Harrington Discovery Institute. I came on about a year ago to be co-director of this transatlantic program where our partners in Oxford in the pediatric department and and our side really synergize, and you'll see in a minute how we do that. I was able to rebuild my laboratory here, and it's just really fully formed now in the Wallstein building, so back to doing kind of original discovery science as well. I think I've told you a lot about what is on this slide, so I'm going to probably just skip it here. Okay, so I guess I can see it here. So the Harrington Discovery Institute, I think, developed in about 2012, had this unique vision of, and I really appreciate this because, there's an approach to drug development that's kind of buried inside pharma and biotech. And the academic physician, scientist like myself, and in fact, I made this decision, what's the best way for me to source that to really become good at drug development? I opted to go learn from the people that are doing it. And it was great experience, and I think it would be hard to substitute. But what they're trying to do here is bring the pharma to the individual's laboratory and really help them develop a drug and get across the finish line, as you'll see in a minute. So it's kind of a unique model. I think there's a bit of this, but I haven't run into very many other examples. And they source within Cleveland, but also you'll see quite widely. So again, really the goal is to accelerate therapeutic development. And this program has evolved such that I think the first effort was really directed at physician scientists. Why would that be? Well, often they are also, the geneticists, the clinicians that see the patients, and they've taken on these disorders to try to develop therapeutics for these rare diseases. It's also expanded to brain health medicines now, and there was an effort in rare disease that has sort of turned into this collaborative effort called the Oxford Harrington Rare Disease Center. You can see here that it has a very wide reach. So every year there are applications called, and I'll show you in a minute. We just sent out the call for the Oxford Harrington Rare Disease Center program. So we get a very large number of applications. The numbers are growing every year. We only have funding for 10 of these every year within each of those domains, mainly the Oxford Harrington Rare Disease Center and the physician scientists. We then take people that are project managers and uniquely the pharma experts that you'll see later to help develop these programs. So the reach is quite wide across the US, UK, and Canada, as you can see. And there's a foundational pre-existing physician scientist program with the red dots, and now these, it's both PhD and MD. You don't have to have both or either specifically to be funded by the Oxford Harrington Rare Disease Center. I'm not going to go into the details of this, but I think it's just an example of what the road is to really develop a drug. This was an individual that was sponsored in 2015 on this physician scientist Harrington Scholar Innovator program. And there's multiple complex steps in this whole process, but excitedly, they're now in phase 3 studies, and this is a medication that is injected before you get to the hospital as you're being transported to save you from NMI. So it's an area of need, and Harrington had a role in facilitating this process. So in rare disease, by contrast, there really isn't, as that was a more common condition, but we have this huge number of things that we're really uncovering. And something that today might look like only 50, 100 cases, a lot of people anticipate that these numbers will go up. Because essentially, as we heard in terms of genetics, it's not standard in that many cases. It's growing, but there are many undiagnosed conditions, I would say, out there right now. And that is really a gap that hopefully will be improved over time. But just in this time window, we have as many as 10,000 cases of rare disease that have been identified. Fewer than 5% have treatments, less than 2% cures. But as was heard, we have some incredibly transformative events like spinal muscular atrophy, which is now in clinic and saving lives. There's the example of cystic fibrosis, where a small molecule can change the conformation of the protein or alter its function. There are opportunities. The convenient aspect of rare disease is you actually know the target. You know what you're trying to treat, and you know what you have to fix. Whereas these mysterious conditions like autism spectrum disorder that are not genetic, we have no clue. We really do not know what to tackle. And surprisingly, there's a lot of human disease that still were in the dark ages, to be quite honest. It affects many, many people in combination. 500 million estimated worldwide. Many are children, but again, it does stretch into adulthood. And some of them are quite severe and take away, cause premature loss of life. So really, now is the right time to accelerate the cures for these conditions. So this is the model that is being used by the Harrington and now adapted by the Oxford Harrington, which is to source breakthroughs within the laboratory and find these individuals and give them the resources that are needed to develop the drugs, de-risk the projects, hit key milestones, which are aimed at therapeutics development rather than the next paper, the next grant, which is kind of not always fully aligned. And the other goal is to understand the complexities of bringing something through the various IND and clinical trial development and finding commercial partners. That's all sort of the attempt there, is to provide at least many of those resources through this program. This, I think you saw, but it just sort of separates out the red dots. There is already within a fairly short time a pretty broad reach of rare disease drug development. And the goal was very ambitious when I joined, which is 40 rare disease drugs in clinical trials by 2034. Obviously, there's a lot of disease that still needs to be tackled, and hopefully this will lay the foundation for how one could accelerate even beyond 40. But there's already, by having brought on Oxford as a partner, already 50 drugs in development, many in the Harrington, but also some in Oxford. And these will kind of help, shine light on the way to bring these drugs to development using these precision genetic medicines. Again, this is the whatever many people have heard is this. There's a promising discovery. We have insight into how, a gene, how the gene is not working properly. How it's impacting to produce the major problems that are arising in the whole, in the individual, whether it's seizures or behavioral problems or gut dysmotility. But then how do you help them get that to a new medicine? So when I was in pharma at Regeneron, what I came to realize is the model is to decide on something we want to do. But then what you typically do is you take that team of scientists and say, all right, stop what you're doing. Now you got to study this. And, and I was thinking to myself at the time, how would we ever get as knowledgeable and expert in any one of these subjects as somebody that has devoted their career to it in Alzheimer's and Parkinson's? So it's almost like there's a disconnect in kind of the people that really understand it, who become, sort of advisors, but in a way, it would almost be a better model to do it within, have those people that have devoted their lives and have a deep, deep knowledge of the subject matter to help them develop a drug. The goal generally is to bridge this gap. There's the Harrington Discovery Institute, which is a nonprofit model, which is kind of fundraising to, fund the individual laboratories, but also to fund the efforts that are needed to get consultants that are pharma experts, as you'll see, the project management and all those other, things that they really need to develop drugs. And then there's a second part of it, which is, the investments that have to be made. There has to be investments made, whether it's starting a company, doing really expensive, early stage clinical trialing, all sorts of things that are very, very onerous, honestly. Doing, safety tests in non-human primates. Those are very expensive things. Luckily, the FDA is starting to lower that bar for us in rare disease therapeutics. Here are some of the domains that really one has in a pharma and a biotech that's really missing in the academic world, which is medicinal chemistry that you can readily access and facilitate a targeted program, specialty expertise in a therapeutic modality. Laboratories take this on themselves, learn it as well as they can, but you can often, at least the pharma will have people that have worked in these modalities for long periods of time and broadly thought about the risks and the utilities. What is the next wave of that a therapeutic agent, nucleic acids, for example? Among the people that are kind of in the know, the first wave is to sort of just do intrathecal, which is nusinersen for spinal muscular atrophy. But the next thing is to deliver it to specific cell groups in the brain, for example. So anyhow, they bring together that pharma expertise. And then, let's see here. This is just a profile of some of the individuals. So there really are people behind this. And many of these are people that have had many years working in a pharma industry, and are sometimes retired. Maybe they've made career changes. It's really an honor to work with these people, and it's just amazing their expertise that's be quite specialized in, for example, medicinal chemistry and further modifications of small molecules, for example. There is a pipeline that's been developed for the Harrington Discovery Institute, and a subset of these are rare diseases. And this is just a couple examples of rare diseases that have percolated through and kind of their outcome. This was for ALS, a rare genetic form due to C9orf gene, and it's a repeat expansion. And this came in 2018. It ultimately turned into one of these nucleic acid genetic medicines programs. There was help at various stages in this process, but it was all done in a way that was sufficient to warrant the startup company Acura Stem. And then Acura Stem was the technology for that ASO was ultimately licensed to Takeda. That's a good outcome because then they have the sort of know-how and the sort of resource to carry it across the finish line to sort of help more people. This is another trinucleotide repeat expansion thing that involves a nucleic acid, but has a broad utility. There was an amazing discovery that these trinucleotide repeats in your body, even in a cell that's not dividing, will expand over time, and it's due to a specific enzyme abnormality. And this discovery within a laboratory I think in Louisiana, uncovered this fundamental key pathway, and it was sufficiently important and valuable that Takeda recognized that and has taken it on. This could act on many things beyond any one of these trinucleotide repeats. And then finally, this is an unusual example, but it shows the diversity of things that we sponsor, is a metabolic pathway defect in mitochondria that causes a deficiency of energy. And people have probably seen CoQ10, in the pharmacy off the shelf. But the problem with it is it doesn't deliver effectively to where it needs to go. So there have been modifications that have been done using chemistry. And this individual, Michael Packold, showed in a single rare genetically determined defect in this pathway, HPDL encephalopathy, that the child that was otherwise, becoming paralyzed and wheelchair bound could walk and even run a bit. So this is one of these transformative examples where you can feed in a metabolic substrate that can sort of overcome a pathway defect. Another example, I think on the left, I'm not going to talk about all these, but IPEX syndrome was awarded the Nobel Prize in 25 because that ultra rare disorder uncovered a key immunologic pathway. The gene is FOXP3, but it's the regulatory T cells that tamp down autoimmunity. And these kids with a deficiency in this have severe autoimmune disorders. We're sponsoring this laboratory, helping them with her big effort to do CRISPR editing of cells ex vivo and other techniques. But, these techniques on this rare disease could help inform a variety of autoimmune disease therapeutic programs. So the rare can translate to common is one of the messages that, I think you should take away. This is the 2024 cohort coming from across the US, UK, and Toronto, so Canada as well. It's a diversity of conditions: oncology, neuroscience, respiratory disease. This is 2025, similar distribution, a lot of neuroscience, but metabolic, oncologic, and respiratory again. And we're just now putting out the application. Deadline is March 9th, and the support is $100K for 2 years, guaranteed, and then this therapeutic development program, as I described. So that's really probably the more valuable component of this. And then once you hit milestones and it looks successful, there's opportunities for further investments in sort of this effort. And certainly they can help with company partnerships and licensing if needed. And so again, the goal is to find projects with rigorous science, innovative programs with clinical impact, MD or PhD from across US, UK, and Canada. You heard the application deadline, and if you want to, you can inquire with questions here or, of course, at this site. And that's it. Thank you.
That was wonderful. I think they were just demonstrating how complex this whole process is, from clinical to therapeutic development to basic science. Well then, Thank you again. The next speaker is Justin Navarro-Marco from the Cleveland Clinic. He's going to tell us about what Mother Nature has taught us.
Something. Hello and welcome. I think this is my 3rd or 4th year being here, and just to take a moment, I know Mitch mentioned this at the start. We talk about rare events. It's rare to get all 3 institutes across the city together, and it's a testament to you, Pam, the rest of the team, and so it's just really special to do this, and so thank you. I get to talk about something, yeah, they do. They deserve a round of applause. I get to talk about something a little different today, a little outside of my expertise, but some of the patients had asked about a talk around biosimilars. And because I'm in the middle and we get a little bit stale, we're going to do a couple kind of raising hand exercises. Who's heard of a generic medication? We could start there, just so we can warm up those muscles. Yeah. Right. It's in the vernacular. We understand maybe what that is. How about biosimilars? We have a couple cheaters here on the side. I know, yeah. These guys might know. How about, yeah, one more time. Who's heard biosimilars, especially from our patients, caregivers? We have a few hands. I would even say many of our clinicians and other folks have maybe a limited understanding of this. I know I learned a lot putting this together. And so we're going to go through this a little bit, maybe using this. I love using the past as kind of a bridge to the future. I talk a lot about that a lot in clinic. Some disclosures, I have relationships with some of the pharmaceutical companies, but we're not going to talk about anything in particular, so nothing too relevant here. We're going to start with a figure. I've been trying to do this a little bit more often. I have zero creative skills. And so like creating a figure is always very difficult for me. So I'm waiting for AI to do it. So I asked it like, what would you do and what would you create? This is the figure that kind of came up with this kind of biosimilars. Maybe a double-edged sword is going to be an analogy I use. Biosimilars have this ability to maybe improve access, help us get patients onto therapies in a more efficient way. But I think there's some challenges with that. And so this figure was created, and I hope to use this. We're going to circle back at the end about maybe the potential of AI. You can see here how many misspellings. I'm not great at spelling, but I even pinpointed and asked it to like respell some of these names, and it could not do it. So I think we're still in our infancy, but we'll kind of end with this AI journey piece, how it's going to integrate into the medical field, maybe this biosimilar world. This is an older slide, but it doesn't really matter. The cost of therapeutics is just exponential, right? This isn't old, it's 2013, but the figure hasn't changed in its trajectory. So medications are expensive, and it's the reason why generics and this biosimilars kind of exist. This kind of data, though, that the idea that the future is here, and I think this was a little bit stark. I don't know if I realized how much this has infiltrated all of our different spaces. These are medications. Many of you guys have heard, this is in the common vernacular. We have family and friends on these kind of medications, rituximab, Remicade, Humira, Lantus. Medications that the biosimilars are kind of leading the market share. And then if we look into the future, and just a few years from now, an explosion in a number of medications that are becoming in this space. Again, medications that we hear about and talk about all the time, and how this integrates into clinical practice, how patients and caregivers navigate this space. Yeah, I think this is humbling. I wouldn't have known or thought about all these medications. So let's start with some basics. And I hate definitions because it's a little bit boring, but I think it helps set the stage here a little bit with us. This innovative reference product, right? This is kind of the medication that we talk about. We can use Ocrevus as one example. I work in the multiple sclerosis space, and it's a big medication there. Tysabri, another. Again, we've kind of understood what generics mean, but this is just a copy of that brand name. We could use Tylenol and acetaminophen. So Tylenol being the innovator, acetaminophen, the generic name for that medication. I think we're comfortable with that. I think maybe something we're also pretty comfortable with, we have a lot of biological medications, monoclonal antibodies that have just taken over. And so a biosimilar is just the generic version of these biological medications, which sounds pretty simple, but I think has great complexity and how to navigate. And I'll have a couple slides, but maybe a picture will be worth 1,000 words to describe the complexity that comes with that. And so we're going to just go right to a picture. We have an innovative kind of product, that reference product. I just wanted to do what it needs to do. I wanted to reduce the disease, right? We either have it compared to no medication or the standard medication, and I wanted to do better. I think this is kind of intuitive. When we talk about it in terms of like generic medications or biosimilars, we're looking a little bit more for equivalence. Does it do just as well as that reference product does? Seems simple, but how we bring that to the clinic is really, really challenging. And so this is the idea that we're trying to shoot for with our generics and biosimilar medications. And I think this graphic is going to do a better job than I could ever do. When we talk about generic medications, like a pill form, right, that Tylenol, acetaminophen kind of question, if you make the molecule the same, we think that the dosage and all the other things kind of come along with it. But a biosimilar, to create bioequivalence, is much more difficult. And again, like a picture is worth 1,000 words. So this picture just shows the structure of aspirin, right? And then you compare it to the monoclonal antibodies. And now we start to see like, maybe not so easy, right? Not so easy to kind of make sure that these products are acting the same way, right? It's just an incredible difference in the structure, the simplicity there of aspirin, many of our other kind of oral products out there. I think in addition to just besides the complexity, these monoclonal antibodies have lots of other elements to them. Again, the details here aren't important. It's just to kind of let you know that there's so much more with these proteins and antibodies that are produced from DNA and a living source and then brought to a patient and infused or delivered in some way. I don't know, it just kind of helps to let you know the complexity that we're dealing with. Because of that challenge, we've had some different regulatory kind of pathways for these medications. So again, reference product I think we're familiar with, though the FDA just changed its path for these reference medications to require 2 clinical trials, usually now going down to 1. So even that pathway is evolving and changing. I think Matthew mentioned some other changes they're making to the genetic kind of landscape as well, and other changes. But that idea of just trying to have some better outcomes makes sense. Generic medications are usually looked at in terms of, are their structures the same? Can we make the dosage the same? And then we're assuming that the efficacy remains there, right? That kind of makes sense. But the biosimilars are so much more challenging, and they want to show that those medications are equivalent at the end of the day. So when a patient with multiple sclerosis, are they doing just as well? And how we define that and how they go through that. It's a different pathway. And so just kind of understanding that regulatory nature has kind of changed given the complexity around some of these biosimilars. And then how do we apply that knowledge here to this group? How does it apply to rare diseases? I don't need to talk to this group about the challenging nature of rare disease. I think we've heard some ideas that it gives us a clear point. We understand some of the pathophysiology there, but it's really difficult sometimes to come up with a clinical trial, a good clinical endpoint, like oncology. Life and death can be the outcome. But when we talk about multiple sclerosis, that's not going to be appropriate. And I think this applies to many of our rare diseases as well. It's hard to come up with a clear endpoint. And so some challenges for these diseases. And then there's we don't have great biomarkers or kind of markers for disease in the blood, CSF, that can really help us monitor how well we're doing. And so I think we have a really challenging landscape. But anytime I have a patient with me, I hope that there's just one message that comes across, because it's a lot of information, but just a hopeful message. I want them to feel this hope. And so I hope some of these slides will help us feel that way, because we've tackled some of these challenges. I think multiple sclerosis is a really good example. That first slide when I showed drug prices astronomically increasing, multiple sclerosis is a great kind of example of that. We've had some oral medications, Tecfidera, Gilenya, the names aren't important, but they're pills that have become generic and have really revolutionized access for some patients. And then more recently, we've had some medications like Tysabri or natalizumab come on as biosimilars, these complex monoclonals that are now available for patients. I don't like the details of the clinical trial for the biosimilar, just maybe brings home this message. You can see that they took multiple sclerosis patients, compared the biosimilar to the reference product, the brand name, and then to see how they did. And patients did really, really well. The medication did what it was supposed to, and the endpoints that they kind of defined. Great, right? It's mission accomplished. I think as this product has come along, it requires a very specific blood test to have to be done. And we've had real challenges in interpreting it, with real challenges of kind of bringing this into clinical practice. And I think it just kind of like highlights that you can go through all these different steps that I just showed and still have challenges at the end of the day to kind of bring this product to patients in a meaningful way. But it's a really nice example of where the pharmaceutical companies have been able to come together to create, hopefully, a more accessible product. And again, this idea has already been kind of discussed, but these biological medications have revolutionized treatment. They're expensive and they're hard to develop. And so these biosimilars have this ability to maybe do it quicker, easier, and maybe more accessible including for those outside the continental US or, our major kind of developed countries. Let's talk about some challenges and maybe some more terminology. People have heard of a placebo effect. We'll do another hand raise. Placebo effect, yeah. The potential positive benefits when we're exposed to something. My wife would say that the placebo effect is very strong in myself, which I'm sure that she has like a really good insight to. She thinks I'm very suggestible. Like a Google review will really kind of weigh on me for a good restaurant. I think she's right. Nocebo. Who's heard of nocebo? It's a weird word. You can raise it proud. We have 1 or 2 people. This is a weirder term. It's kind of the inverse, though. So if I tell you that you're taking a generic medication that feels maybe second rate, not the same as that brand name, that can also do the same thing, just in a negative way. Nocebo effect. Well described, well established in the literature, not so much in a common vernacular, like I never use that in the clinic, has had huge implications in how patients and clinicians and others have thought about these generic medications and biosimilar medications. And just like we talk about placebo in a scientific way, I think we can talk about nocebo effects as well in a scientific way, but just understanding that that exists is really important. Many examples that this could be applied to, but this is one of the challenges that we've had to develop that just the notion that it's not that reference product makes it sound maybe less good and has less efficacy. I struggled with how to present like a well-balanced talk on this topic. Who's read this book, or maybe who's read it? I forced the one person in the back to read it. Has anyone heard it before? Yeah. My wife and I were on like a car ride. We heard it on a podcast, and then we picked up the book. It was an investigative journalist who talked about mainly generic oral products and how that kind of journey has gone on. And again, I lived this world, and I didn't know much of what was kind of presented. And again, it's just one side of the story. She does a really nice job, though, diving into how the industry has expanded beyond the continental US, that much of the manufacturing for some of our generic medications is done outside the continental US, mainly in Asia, and talked about the challenges of the FDA being able to monitor things in the continental US, and then you try to do it outside of it. And you can understand some of the challenges that come with monitoring that world. So I just wanted to present maybe a little bit of this. Sure, nocebo effect exists. When people think that they're not getting that reference product, I think that creates challenges. But I think she does a really nice job illustrating that some of those challenges are real, and it's really difficult to disentangle. And this paper just came across my email, and I was like, God, that was timely. Again, we're still in this multiple sclerosis. Gilenya is a pill, so it's not this monoclonal antibody, but just looking at this as a medication that is just starting to kind of get into clinical practice, we're seeing some concrete differences in disease outcome with the reference product versus our generic meds. And then this table was just like humbling. The details aren't important. It just shows that there are differences in all the different versions that are out there. To me, to think that none of those things have some impact on what's going on, I'm not sure. So we can talk about the nocebo effect, but we can talk about this other element as well. And I think we still have lots to figure out here. I said that I would kind of end and talk about maybe like a story of hope, and I'm hoping to leave some time for questions. I just learned this. Who's heard of Keppra? Levetiracetam, yeah. Revolutionized the field. I didn't know its backstory. 1960s, they were thinking about it as helping to improve memory and cognition. You can guess how well that went, because no one's heard of it in that sense. In the 1990s, the pharmaceutical company was just trying to look for new and innovative ways to find things, and they found that Keppra had this incredible benefit for seizures. And all the seizure medications before this, they were harsh, terrible side effects. And this medication brought forth this like revolution in how we think about it and new products that we still use today. Keppra is my go-to medication. And I think that when I think about AI, to be able to go through that database of things that the pharmaceutical company already has or scientists, to me, we're going to see these kind of things recycled and reused and hopefully have these different opportunities. But I was just thinking of like medications in general, that the way that we think about them and discover them, it's just going to be huge changes. I just want to take a step back and just kind of like help lay the land for what we kind of talked about. And again, I don't have all the answers. Like, what does a biosimilar mean for individuals? But I hope that people feel a little bit more knowledgeable. I certainly did not know some of the elements in this talk, the way that this is already kind of infiltrated into clinical practice. And I'll tell you that even as a frontline clinician, when I'm ordering a med, it's not entirely clear which medication version you're always ordering. And so it can be really hard, but now I hope that we can empower patients and caregivers to at least ask that question, understand what that term means. We have generic medications, we have biosimilars, not better or worse, but they're different, and they have different pathways in which they're approved. The current pharmaceutical landscape is unsustainable, and there'll be lots of changes coming. This will be a part of the solution, no, but we can be part of the conversation and then understand some of the challenges that come with that. And so I hope that I've done that and not upset too many of my pharmaceutical colleagues that are here. And yeah, I'm happy to take some questions. And yeah, thanks again for the opportunity to come.
So it's striking the molecular size differences is striking that you showed. The antibody and aspirin.
Yeah, whatever it is.
So do you think that that's one of the limitations and maybe why that newer drug is showing greater effect in MS, because it really reaches areas that the antibodies don't?
Yeah, I'm not sure. I think, to me, that like the difference is just like humbling to know how complex the pharmaceutical company has to go to order to recreate those products. And it makes sense that there are different pathways. I don't know if we've seen like clear that like a biosimilar is going to be better or worse than some of the other medications, but different, I guess, in my mind. And I'm not sure what that holds. Like, what does that mean for the future?
Other questions?
Families or caregivers and how they approach that question, I don't know. Yeah, I'll take any. I think I have 2 minutes. Okay.
Pam?
How do you know if you're receiving a biosimilar?
I mean it when I say that I'm not always sure.
The question
think-
is, how do you know if you're receiving a biosimilar? So,
You click a button within Epic on a medication that sometimes just says, we can use rituximab as a good example. But then in the background, there are like 4 or 5 different products that are available. And we don't always have an opportunity to think or know which one your insurance company has kind of already had that relationship with. It's 100% fair for patients and families to start asking that. And again, I think the importance for me, there's a whole team behind me. I have a pharmacist who specializes in this world, and we can have those conversations. But I think coming with a little bit of power and knowledge around it is important. And it happens so fast in the background and usually made at levels well beyond the individual patient encounter. Administrators, insurance companies, they're making these decisions on where these kind of medications go. And again, has a tremendous opportunity. I hope that I can present like this balanced approach. There were people that I could never get in a monoclonal antibody that with the biosimilar, rituximab is now almost universally, like if I click that button, I can usually get a patient on now. NMOSD is a great example where 5 years ago, that was not the case. The biosimilars have revolutionized that. But I think it comes with a double-edged sword that I'm going to try to be a little bit more aware about in my practice. So it's a long-winded answer to say, really requires some awareness around some of the changes in landscape that we have to navigate. Okay.
thank you.
Please.
5-minute bio break, coffee refill, and then we'll get right back to it. Our next presentation is remote. It's Allison Bradbury from Nationwide in Columbus. So we have Allison Bradbury from Nationwide Children's in Columbus and Ohio State University. Allison is going to talk about translation of genetic therapies for rare diseases from bench to bedside. And please bear with us as we get the sound and all adjusted. Allison, do you want to, if she can hear me?
Great. Thank you very much for having me today to join Rare Disease Day in Cleveland. My name is Allison Bradbury. I'm sorry, I can't hear the room, so I don't know if this is already introduced. But I am a principal investigator at Nationwide Children's Hospital in Columbus, Ohio, an associate professor in the Department of Pediatrics at Ohio State. And I'm really pleased to be here today, so thank you, Pam and Mitch, for allowing me to join. Sorry I'm not there in person. But today I'm going to talk about translation of genetic therapies for rare diseases from bench to bedside. Okay, so today I'm going to level set and go back to the central dogma of biology, where most of us in the room know that our DNA is transcribed into RNA, and RNA is translated into proteins. This instruction of our cells is read in an all-important 3-letter code known as the codon. So each of the 3 letters of instructions in our DNA and RNA then indicate what is the next amino acid or the bead on the string of the chain, which then provides the full instructions for our functional proteins. Just a single change in a letter of our over 3 billion base pairs of genetic information can really impact the structure and function of our protein and result in rare diseases. So some of these changes to our DNA may result in a protein being completely degraded, while others may result in a protein that is shortened, or some changes in our DNA may result in a protein that has a different structure or function. Once we really understand how changes in our genetic code impact the function of our protein, then what my lab focuses on is the development of genetic therapies. So can we really go to the root cause of the disease, those changes in our genetic code, to alter the course of rare disease? And one of the ways we do that is with what's called gene therapy or gene replacement therapy. So oftentimes these changes in our genetic code result in the loss of function of a protein. So that protein is degraded or short or not able to do its job to its fullest capability. In that case, we can use gene therapy, in which we use an adeno-associated virus gene therapy, or AAV for short. We've already heard today about the example of Zolgensma for the treatment of SMA, and this is the same type of gene therapy. We use this virus, and we put a gene that has a variant in it causing disease. We put a healthy functional copy of that gene inside the virus. The virus then gets inside of our cells and deposits a functional copy of our DNA, which then makes a functional protein. So this does not interact with the DNA that's already in the cell, but instead is providing a new functional copy. So this is why it's called gene replacement therapy. So today I'm going to give you 2 examples of a bench-to-bedside translation of a gene therapy that I worked on in my lab, and I chose 2 examples that both had implications in Ohio. So the first story I'm going to tell you about is globoid cell leukodystrophy, also known as Krabbe disease. Krabbe disease is a lysosomal storage disorder that results from the deficiency of an enzyme known as galactose reductase, or GALC for short. In the deficiency of GALC, there's an accumulation of a cytotoxic substrate known as cytosine, When cytosine accumulates in the all-important myelinating cells, the oligodendrocytes in the central nervous system, and the Schwann cells in the peripheral nervous system, this results in widespread central and peripheral nervous system demyelination. Krabbe disease has been identified for over 100 years, and the classic infantile onset is typically fatal by about 2 years of age. For Krabbe, the standard of care since the 90s has been hematopoietic stem cell transplant. And what we know about hematopoietic stem cell transplant when babies receive umbilical cord blood from healthy donors is that if the babies are transplanted early in development, this can improve the overall survival in this disease, and it can help with cognitive neurodevelopmental outcomes. However, the transplant does not help the gross and fine motor that really is one of the hallmarks of Krabbe disease, because it has that both the central and the peripheral nervous system demyelination. And what we know now is that the transplanted cells do not efficiently target the peripheral nervous system. So for our first gene therapy approach, what we wanted to do was to retain that standard of care that we know does have some benefit to the central nervous system, but add on an AAV gene therapy. So what we do with gene therapy in the lab is that we choose the AAV capsid, or that shell, and there are multiple serotypes of AAV, including AAV9 and AAV-RH10 are some of the serotypes that we know can cross the blood-brain barrier. Once we choose our AAV serotype, we then put in the genetic material that we need to replace for that disease. So for Krabbe, it was the GALC gene. And in this case, we delivered the gene therapy intravenously because we really needed to hit those peripheral nerves. And what we know is that even when it's given intravenously, some of the gene therapy product can get out of the bloodstream, cross the blood-brain barrier, and enter the cells in the central nervous system. The virus then deposits that therapeutic transgene, and the virus shell itself is rapidly degraded. The cell can then become like a factory producing that therapeutic protein. So in this case, then the GALC enzyme started to be produced by the cells. And we have an extra advantage in that this is a soluble secreted enzyme, so it can even get out of these cells and picked up by neighboring cells by receptors on the cells. We do know that with gene therapy, especially with neurodegenerative diseases, the cells start to die off with the disease process. That's why we know we need to intervene as early as possible in the disease process with gene therapy while you still have a number of those healthy cells to receive the virus and become those all-important factories for your therapeutic protein. So with Krabbe disease, we had a lot of advantages and that we had really important model systems had already been established. And one of those was a preclinical model that is a naturally occurring mouse model known as the Twitcher mouse. So I'm not going to go into any of the real details of these preclinical studies in the interest of time, but this study was testing the transplant and gene therapy in the Twitcher mouse. This was done by David Wanger's group. And what they were able to show is that adding gene therapy to the transplant had a synergistic impact, and that there was a clear dose response with the gene therapy, meaning the more gene therapy they gave, the better the mice did. I was at University of Pennsylvania at the time and had the added advantage of having a large animal model of Krabbe, which is a naturally occurring dog model. So again, in the interest of time, I'm not going to go into the details of this published study, but I will just say that we recapitulated the study done in the mouse, but in this really valuable large animal model. And we also were able to establish that there was a combinatorial or synergistic approach by adding the gene therapy on top of the stem cell transplant. And we also saw that really important dose response with the more gene therapy that we gave, the better the dogs did. So I know it can be a little bit difficult to use large animal models in research, but I do just want to say that they really are valuable because they can really recapitulate many of the symptoms that we see in human patients. So you'll be able to notice in this puppy that has Krabbe disease but has not received any treatment that it has a wide base stance and has difficulty ambulating. This dog also has a tremor phenotype and will progress to lose hearing and vision. This disease in the large animal model will eventually lead to pelvic limb paralysis, which is our humane endpoint, and that occurs by 3 to 4 months of age. So just like in the babies, the disease is really progressive in this large animal model, and it gives us that therapeutic window to intervene. And so then I now would like to show one of our treated animals that received the gene therapy and the transplantation, and this dog is 2 years old. So we've significantly increased the lifespan of this dog, but this dog is also neurologically normal at 2 years of age. So you can see this dog can play fetch. This dog can play fetch and can also has normal MRI and nerve conduction velocity at this time. Okay, so at this point we were able. Okay, I'm getting some audio feedback right now. Do I need to pause? Okay, I guess I'm gonna keep going. I'm hearing the media vision person on the audio. So at this point in the Krabbe dogs, we were able to show that transplantation and the intravenous gene therapy were able to correct the central and the peripheral nervous system demyelination. And then we did the necessary studies, the safety and toxicology studies, to really translate this to the clinic. We were really excited about these unprecedented results in this really devastating disease. And fortunately, we were able to license this to a company in Columbus, Ohio, Forge Biologics, who initiated a clinical trial. At this time, Krabbe was also added to the newborn screening, which is really valuable to be able to pick up these babies at birth and intervene as early as possible. So with this clinical trial and Krabbe being on newborn screening, 6 patients have been treated to date. But I will say that Krabbe was just uniquely poised for this success because there's this rich 100-year history. We've known about Krabbe for a really long time. The mechanism of disease was really well understood, which enzyme we needed to replace, what was really damaging the cells and causing the disease pathology. As I showed, there was multiple preclinical model systems that were already available. And since the late 1990s, there was a standard care in human bone marrow transplantation. Beginning in the early 2000s, in that mouse model, the gene therapy began being developed. So what I showed you today were all studies that happened after 2020, but they were really possible because of this really rich history of the understanding of the disease and the generation of model systems. But what happens when now with the advances in really genetic medicine that we can identify disease-causing genes much more often with all of these over 7,000 rare diseases that we have. We don't have 100 years to wait to develop therapies. So one program in my lab that I've been involved with the last 5 years was the development of a genetic therapy where I partnered with SLC 6A1 Connect. And we kept this as a complete academic and foundation partnership. And we were able to do our first in-human gene therapy in September of last year. So we're really excited about this and possible ways to develop and translate genetic therapies faster, and really just want to highlight the importance of patients and foundations in advancing many of these rare diseases. So Mitch mentioned this at the beginning, but there's already been advances this week in advance of Rare Disease Day, and the FDA is trying to make some of these pathways and processes that can expedite the development of gene therapies. So they just put out the draft guidance on the 23rd and be open for comments. And we really hope that implementing some of these practices at the regulatory level will improve the ability to implement gene therapies faster. But I will say I sit on the board of directors for the American Society of Cell and Gene Therapy. We have a lot of patient-friendly resources, educational resources. So for example, you can watch Gene Therapy Basics. We have little videos and also pamphlets. We also have an Empowering Patients Summit coming up on March 11th and 12th, if that would be meaningful to your community. And we also have a patient advocacy newsletter that comes out called the Patient Press that has a lot of really valuable information for rare disease communities. So thank you for your time and for letting me be involved in your Rare Disease Day in Cleveland. And I think I will try to stick around for questions if we're able to get audio to me from the room. So thank you very much.
Questions, I will send them as text to her. Anybody have any questions?
I can hear you a little bit, Mitch.
I'll try again. Any questions? Now you hear me, Allison?
Yes.
Okay. No? Oh, go ahead, Matt.
So,
Yep.
there are newer methods to facilitate delivery of AAVs. You had serotypes, but also even linked to transferrin receptor antibodies, for example. How do you see that progressing in the future in terms of targeted delivery of AAVs?
Yes, that's a great question. So there are definitely some next generation AAV capsids that are being developed right now. And one of those that I think will likely be one of the most successful is the transferrin receptor targeted capsid. It's being used in a lot of preclinical studies. That specific one hasn't made it to the clinic quite yet. It's difficult to model some of these capsids in preclinical studies because it's a human transferrin receptor. So while we can get some data using this like humanized receptor mouse, I really think the first in-human clinical studies will be really informative for these. Some people might have heard that in September, there was a novel capsid, a novel CNS capsid by a company called Capsida that was delivered to a child with STXBP1. And unfortunately, that child passed away 3 days after the gene delivery. So I think there's some excitement around these novel capsids, but we also need to be cautious and recognize that there will be a first in-human delivery and that the risks of those will be unknown until they're actually tried in human patients.
All right. Thanks, Allison. I know you've got a
Yeah.
conflict, so wonderful. Sorry for the technical issues, but that was a wonderful presentation. All right. So next we have Tamia Woods, and I'm going to let you introduce yourself.
All right. Well, first, I want to sincerely thank Ms. Pam, Dr. Drum, and Dr. Abeta Marco for inviting me to speak today. It means a lot to be amongst you all, especially for rare diseases and something that is not so common. You make patients feel seen, and I'm honored to be in front of you today. So my first question, raise your hand if you think that I came from Rochester here, since I work for Mayo Clinic. I actually live here. So my name is Tamia Woods, and I am your clinical specialty representative for Northeast Ohio and for the neurology department. My talk here today is from complexity to connection, supporting providers for rare autoimmune neurology disorders at Mayo Clinic. And at Mayo Clinic, this is actually my team. We are all over Ohio. And we actually have what we call CSRs, but they're clinical specialty representatives that are positioned all across the United States and also in some countries internationally. We are an extension of the neurology laboratory and other allergies as well, and serve as also a liaison between the consultation teams. We take very much a large amount of pride into making sure that we keep providers informed about the latest discoveries on autoimmune antibodies and emerging diagnostic testing. The field is evolving rapidly, and our responsibility is to make sure that you all have access to the most current information that is clinically relevant as well. So in neurology, we say time is brain. And I use this as a visual. 50% of patients with NMOSD, if left untreated, will be wheelchair bound or blind. 1/3 will have died within 5 years within the first attack. And there are 4 therapies that are approved by the FDA since 2019. And that is why the sooner we can help you reach clarity, the sooner your patients can begin treatment and on the right healing journey. So over the years, we had in 1960 one antibody. And we have discovered more than 50 since then, including Septin 5 and 7 that were recently rolled out and actually discovered by Mayo Clinic. But with that, we have evolved far beyond the perineoplastic evaluation that we created in the early 2000s. And although it was great at the time, we now have phenotype-specific evaluations that are available to you. And the reason why I put the perineoplastic evaluation at the bottom is because I want to give you a full visual of where we came from early 2000s to the actual phenotype-specific evaluations that we have here. And if you are ordering a test and you just ordered the perineoplastic, how many antibodies you are actually missing on your actual patient. And it's very important for that. Number one, I, as a clinical specialty representative, is doing a disservice for you if you don't know about the perineoplastic evaluation. Also, you begin as providers to create your own evaluations, ordering 2 and 3 different tests to get one that we already created for you. And so I just want to make sure that you all are aware of all of these tests that we have. And I actually have them if you want one to go with you today. But my role is to be more of a liaison for testing. I aim to be a reference and a resource for you. Mayo supports that commitment by offering podcasts, webinars, micro-learnings, and so much more. I created this QR code for you to scan, and it takes you directly to a website that's a learning tool. So not only are we doing the research for you, we are also making sure that you can go to these particular websites and access them on your time, because I know we are so busy. Beyond that, we have consultants, and our consultants are here for you. So unlike any other laboratory where you're just ordering tests and sometimes you may not know what to do next, we have consultants that are available for you that would be able to not only guide you before you order the test, during and after. We also have opportunities that you can actually schedule through me where I can organize a discussion where we have customized question and answers. I can personally train you on certain topics as well. And it's also available to residents and fellows. If you ever need guidance, you can always let me know. But my main thing and Mayo's main goal is to make sure that you all are aware that you don't have to navigate through these complex issues alone. We are there for you. Finally, I wanted to let you all know that I, along with my Mayo neurology team, will be at the American Academy of Neurology in Chicago this year. And I hope that you all stop by our booth if you actually attend. There will be consultants as well, so we can continue this conversation, but also I can introduce you to those consultants. Lastly, beginning on March 23rd, I personally will begin to send out newsletters highlighting some quick updates. You can have access to our awesome digital algorithms, and it will also have other need-to-know information on there. I'll be around afterwards where you can sign up for my newsletter. We can continue on with these discussions. And also, if you have any questions, I am around as well. It is truly an honor to support you moving from complexity to connection. And I'm looking forward to working closer with all of you. Thank you for all the work that you do. I appreciate you.
question in. And then you can accost her during the lunch break.
Yes.
Thanks, Timmy. All right, so let's move on. Our next speaker is Tracy Marie Greenberg. Tracy is going to talk about the self-advocacy ripple effect.
Hi, I'm Tracy Marie Greenberg. It's an honor to be here. I'm part of the planning committee for Cleveland Rare Disease Day and also a rare disease patient. And I'm here to talk about the self-advocacy ripple effect. Ultimately, what I'm going to show is how self-advocacy can lead to cures and treatments. There's a lot of other things that self-advocacy can create a ripple effect in the community, but ultimately that's our goal as patients and advocates, that we want to see treatments and cures. And I'm a patient family advisor at Metro Health, and I'm also on leadership council at the Cuyahoga County Board of Developmental Disabilities, where I receive services. And patient advocates are anybody that's a family member, a friend, a caregiver, a person with a condition, and sometimes the medical community can advocate for themselves if they are passionate about different situations and people that they're caring for as well. And you can see, I'll go ahead and describe this for low vision individuals. The image before you is of a person's hand disrupting water, creating a little bit of a ripple effect in the water. And on this next slide, just to describe, is a person pulling on a string from what would be considered maybe a ball of yarn or a big shape, but they're pulling on the strings, and so they're hopefully being an advocate, and that's what advocacy requires, is that we pull on strings and levers and all kinds of things. But what we're doing essentially is a whole bunch of stuff as advocates. It's almost a full-time job. So you're gonna, ask and answer questions, and sometimes people don't speak up for themselves, or they're afraid to ask questions, or they get nervous and forget what questions they're going to ask. But an effective self-advocate is going to probably come with a binder, and you're going to be a little overwhelmed, and hopefully they're going to make a great partner with the medical community. Giving opinions is very important. There's a very big spectrum of effects when you have a rare condition. So giving that information to those around you is very important, because you're the only one a person who's experiencing something is the only one who can really understand the information that they're trying to process through what they're experiencing. And learning and doing research. And I know that we're always being told, don't Google it, don't Google it. But I've had providers say, I'm sorry, I just had to Google your condition. And I appreciate that, because if you know how to look through the information and you know how to read between the lines, then it can be a valuable way to learn about your condition. I did get scared when I first Googled. I was late diagnosed. I was diagnosed at age 42. So I did get a little bit scared when I Googled my condition, because there was mortality things and things that might not necessarily be affecting me, because I'm on one end of one spectrum, and I was seeing all of the classic types. So that brought a lot of questions. And keeping records, as I mentioned, sometimes we show up with a binder. Writing letters. Writing letters is a very effective way to communicate. Sometimes, we forget what we wanted to say. We might have a list, but you could follow up with an email. Hopefully your providers are reachable. And writing books is a huge way to create a self-advocacy ripple effect, because others can read that book and gain insight and inspiration through others' experiences. Creating community. Here we are today because of patient advocates. A group of patient advocates got together and said, we want to create an event in Cleveland, and it started out very small, and it's exploded into a great event with all of you fine people here today, so thank you for being here. Thank you to Pam Judge, our top patient advocate here, helping to coordinate this whole thing, heading it up. And spreading awareness is really important. That's what I'm going to be doing here today for my condition, as well as all the other things that we've discussed today, creating a greater understanding of what the rare disease community is facing as far as challenges for arriving at the treatments and cures destination. And starting a nonprofit, we have a lot of parents and families and individuals that are passionate and they want to make something good out of something bad, so they will start nonprofits and to further their own mission of finding treatments, cures, and to provide services to people who are experiencing these situations. Joining boards and commissions. I myself am on a whole lot of boards and commissions. I'm on the Patient Family Advisory Commission at Metro Health. I'm also part of the Rare Disease Day Planning Committee. I'm on Leadership Council at Cuyahoga DD. I'm also on several commissions at the City of Lakewood. I was the chair of the ADA Transition Plan Task Force, so I headed up a mission for creating and improving accessibility for those with mobility and visual and such challenges. Organizing fundraisers and events such as today. Participating in panel discussions, and we're going to have a great panel discussion here in a few minutes. And most importantly, partnering in the development of treatments and cures. And that partnership is this next slide. At the top, it says partnerships in rare disease, and then there's a big blue double-sided arrow pointing between the groups of partners. Sometimes there's crossover. Sometimes the researcher is a patient, and that's why they're passionate about that cause. But mainly they're patients, caregivers, and parents who are the self-advocates, and then they're interacting with the medical community, the researchers, the administrators, the providers, educators, institutions, including pharmaceuticals and nonprofits. And the commonality and the common goals below are also with a graphic, a blue graphic of a thought bubble, desires for answers, and above inspire innovation is a graphic of a light bulb. And developing protocols, there's a check mark, a blue check mark, and ultimately treatments and cures, and there's a little picture of a medicine bottle. And on this next slide, this is the only one I designed without AI, so you could see it's a little bit plain, but I did want to include the Rare Disease Day colors, which are purple, blue, green, and pink. And at the top of this slide, you could see a heading, Rare Disease Self-Advocacy, and then there's the two parties that are communicating with each other are the people that are living the experience. And then you have the people who are learning the experience. And the people living the experience, this is their everyday 24/7 experience, so they know it better than anybody. And then you have the support community who is learning the experience, our doctors, the researchers, the caregivers. And the questions aren't always asked, but an effective self-advocate is going to be answering or providing information that might answer the following questions, such as, what's it like? And each individual has their own lived experience. What have you learned? What are your challenges? And what are your needs? And if we're doing a good job at advocating for ourselves, we're answering these questions, or we're offering this information, we're delivering it regardless of whether we're asked. And there's an image of a gentleman using a wheelchair speaking with what appears to be a provider in a hallway of possibly a hospital. And he is the self-advocate, she's the provider. And they're having a discussion. She's showing him her computer. She's showing the computer screen to him, so she's explaining something. And the self-advocacy ripple effect, just to overview, The results will transform personal growth for the individual who's doing the self-advocacy. It creates systemic changes. It benefits the individual as well as future generations. It leads to customized effective care plans. It can create specialized care teams. It inspires innovation and research. And as mentioned earlier, creating community and systems of support, as well as resulting in treatments and cures. And on this slide, on the left is a picture of what appears to be possibly a patient with a parent, I'm guessing. And parents are the greatest self-advocates for their children. And so there's nothing more powerful than a parent who's looking for help for their child. And then that creates the big wave. The ripple turns into a big wave. And so that's what we're here for, to create that wave so that we can find better treatments and cures. And then I wanted to just take a moment and show how the crossover can go between self-advocates and the medical community. You may have heard of the doctor who cured himself, Dr. David, I assume Feigenbaum is how he pronounces it. And he's a best-selling author of the book Chasing My Cure, A Doctor's Race to Turn Hope into Action, and a patient battling idiopathic multicentric Castleman disease. Hopefully I pronounced that right. There's a picture of him on the right showing him athletic, healthy, and he's a medical student. And there's a picture of him with a stethoscope dressed for his headshot. And but Dr. Feigenbaum was diagnosed during medical school with Castleman and decided to try to figure it out. And there's an image here showing him battling this illness, and he has lost his hair, doesn't look very healthy. He's taking a selfie in a mirror. And we're just going to go over the bullet points on the slide showing how he cured himself. After being diagnosed with Castleman disease, he became an expert in his own condition, which had been poorly understood at the time. He studied his own blood samples, and he developed theories about a potential treatment. He identified a drug that had already been approved for transplants that targeted a similar cellular pathway that was active in his disease. And he then worked with his doctors in order to get that drug approved to repurpose it in his treatment. And the treatment put him into complete remission, a state that he has maintained. And as I mentioned earlier, people do these, they become passionate about something for a reason. He obviously became passionate about treatment of rare conditions, because you could see this next slide shows him more recently, I'm assuming. He looks happy and healthy, and his list of credentials reflects that he is now working in the rare disease space, helping with orphan drugs and new discoveries in rare diseases. I won't read all of his credentials, but you can look it up. And Tiffany mentioned that she met him, so he's a very nice guy, she said. I didn't know that. I knew that he was nice, but I didn't know you met him. So I'd like to introduce you to my friend Sherry Lube, and she is a patient advocate, self-advocate. She's one of my favorite patient advocates. She's a really fun person, and she's highly involved with the National MPS Society, and her shirt is awesome. This picture of her I saw on Facebook. I'm like, oh, I'm using you in my presentation. Is it okay? So I had to get that shirt. The shirt is a picture of a zebra unicorn hybrid, I guess. And it says, I confuse physicians on her shirt. And also she has a sticker that says, I voted. And a list of her things that she does. She's on several committees at the MPS Society. including she's a member of the Board of Directors, Education and Communications Committee, the Advocacy Committee, the Family and Adult Resource Committee, the Fundraising Committee, Family Conference and Planning Committee, as well as she meets regularly with lawmakers in DC. And Sherry and I both have the same rare disease called Morquio syndrome. It's a form of MPS, and we both have it. It's type 4A, and it's, as earlier mentioned, lysosomal enzyme storage disease. This is a similar type of disease as the Krabbe condition we heard about before. And the enzyme, the Gallon's enzyme is the one that's missing. We commonly have dwarfism. As you could see, we're all different heights, so that's the spectrum. You know, we all have the same condition. And I was late diagnosed, so when I first went to meet the MPS community, people kept saying, "Oh, you're so tall," which was funny because I'm only 4'9". I'm like, I've never heard this before. So, and it also can cause, it causes dwarfism, skeletal manifestations, respiratory challenges, in some are vision and hearing issues, kyphoscoliosis, and spinal cord vulnerability, including also muscle weakness from that spinal cord vulnerability as well. Hopefully I can do this because she has a little video that she made for us, and I'm hoping this works. Do we know how to play it? If you just click on it. You have a mouse there? Is this a PDF or is this the actual PowerPoint? Oh, there you go. I was a little worried about this. Oh, there she goes.
My name is Sherry Wu. I have a rare form of dwarfism called Morquio syndrome. Right now, there are only about 3,000 people in the world with my diagnosis. Every year, my husband and I travel to Washington, D.C. to talk to our elected representatives about why rare disease research is so important. You don't have to travel all the way to our nation's capital to make a big impact, though. You can always talk to your local and state representatives about why rare disease advocacy is so important. Get together with your friends and family. When we all band together and make a big group, we make a bigger impact. And remember, only the squeaky wheel gets oiled.
So that's what we're doing. That's what we're here today for. So we love Sherry. She's so much fun. And she also receives Vimizim, which is the enzyme replacement therapy for our missing enzyme that we received from BioMarin. And when I first entered this space and knew that I had Morquio syndrome and knew that I was going to receive this treatment, she was a huge support for me. So that's the community. That's the community. She said, I said, should I get a port? She says, I love my port. I said, this doesn't make sense to me. But when I got my port, I said, I love my port. So, and finally, where's the last one? We have one more slide. It's missing. Okay. Well, the last slide was an image of Judy Heumann, and she is considered the mother of disability advocacy. She helped make the ADA come to reality, and she's just a huge inspiration in the self-advocacy world. Unfortunately, she passed away recently, but I have a quote here from her. I did have a picture of, I don't know what happened to that last slide. I love this quote from Judy, and I use it all the time because this is how it feels to be a self-advocate. Change never happens at the pace we think it should. It happens over years of people joining together, strategizing, sharing, and pulling all the levers they possibly can. Thanks. Thank you.
Sorry, I think we have to move on here. So our next speaker is Eric Feingold, and Eric is going to tell you about a topic you think you know something about, but having a different application than what folks are typically aware of. So Yeah, go ahead.
Hello?
An on button. How did I do? Okay. Okay, we said fireside chat. There's no fire, but it's warm in here. Thank you for being here. Thank you to Pam and Pam Judge and of course Dr. Drum for all the effort to put this day together. It's an amazing event and I'm glad to be part of it. I'm Corey Wolf. I am Director of Patient Advocacy for Patient Health Alliance, formerly Guthrie Jackson Charitable Foundation, and a caregiver to a daughter with a rare disease who was diagnosed about 10 years ago after a very difficult and long diagnostic journey. I'm on the planning committee for this event, and when we were talking about sessions for what we should be talking about today, I just kind of threw out there, maybe we should bring up AI. And there was no reaction. Nobody wanted to raise their hand. They're like, do you know anyone that could do that? And you know, these are brilliant people we've heard from today. And Dr. Kaplan was lovely enough to introduce us to the team at FDNA. And we're so glad to have Eric with us today. Do you want to introduce yourself first?
Sure. Hi, I'm Eric. I'm from FDNA. I'm the CEO and co-founder. By education, I come from game theory. It doesn't mean that I played Doom or Space Invaders in college. If you know the names of those games, you dated yourself. If you don't, you may be too young to be here. By experience, I'm an entrepreneur. I had the good fortune of establishing a number of health tech companies, including a company that can extract health biomarkers from the voice signal. So determining everything from neurological conditions to behavioral health. And here with FDNA, it's the ability to detect critical phenotypes by analyzing soft tissue, all using AI, of course.
This is amazing. I had the privilege of meeting with you previously and learning a little bit about this, and I've been diving in ever since we met. So I just want to start with some questions because I know that there's a lot of skepticism when we talk about AI in medicine. But how can AI-powered phenotype recognition support clinicians at the moment of clinical suspicion, both in general pediatrics and pediatric genetics, integrating into existing workflows to reduce data and time burden while augmenting but not replacing clinical judgment?
Sure. So there is something about the word AI or the name AI that works against it a little bit. It suggests a form of alternative intelligence that competes for the decision-making process about anything. And really it is not. So AI is at its best when it is used to take enormous amounts of data, unstructured data, data that is sometimes very difficult to get in the first place, and then creating a structured set out of it that could be used for real decision making, and then perhaps get insights that can dramatically accelerate the decision-making process, especially in complex conditions such as in rare disease. So think of AI as something that can take a mountain of data that you could probably do on your own, but it would take you hours, many hours. In the example, for instance, in our work with Weill Cornell, where our system is used, they said that that a typical review of a genetic case can take somewhere between 8 to, I don't want to use the wrong number, but between 8 and 16 hours or so. Like, it's just very, very long. Whereas using the tech, you can do everything in under an hour or, actually in just a few minutes. So if you extrapolate that to thousands of patients, to hundreds of patients, you basically get the difference between being able to offer care and research and therapy and diagnosis to many patients or to just a few. That's where the AI makes a real difference. Now, if you look at the latest, I just want to take one example. If you look at the AAP guideline from July 2025, that said that any child with general developmental delay or AD should undergo phenotype first testing. If you think about this guideline as a guideline, I mean, it's very well intended, and it could really make a huge difference. And if we have time, we can talk why this can be such a huge deal for all, because it can bring the critical mass. It can reduce the number of underdiagnosed, of misdiagnosed. It can or completely undiagnosed. But in practice, what it actually means that a pediatrician that encounters during a regular check someone who, where something doesn't look right and it looks like a developmental issue, that pediatrician actually has to make a choice, to send that child to a deeper review. But to do that, they need to collect a lot of information and to create a well-structured clinical note and maybe document some of the phenotypes and do all those things. Now, the fact is that it's not going to happen, and it's not because they don't care, but because pediatrics, general pediatricians or general healthcare is really built for speed. It's built for population health. It's not built for the special cases. And the truth is that even getting reimbursed for that effort for the doctor is a real challenge. So, but there is a way to break that. This is where a phenotype, where a digital phenotype AI that can essentially do the work within seconds can make the maximum difference, because that can actually make it possible. So what you would get out of that is the ability to move some of the work that would otherwise be in the hands of a geneticist or a GC much later in the process. It's an opportunity to move that work upstream to the pediatrician. And that means that the GCs and the geneticists can focus on things that only they can do, right? Essentially, solving part of the problem, part of the bottleneck that exists in this space. So this is in a nutshell, but I think it will be easier, if that's okay with you, that if we'll show you just in a couple of slides what the technology actually does. It will give you a tangible sense of what it means, because without it, it sounds all too theoretical. So if you see it, I hope it will make a difference. Yeah, would you mind? Good. So first about what we do. So this is the case with FDNA's AI. What you would have is you will take a picture of soft tissue, or specifically, in many cases, the face. And the technology is able to recognize and see things. So this is the kind of visual intelligence, the kind of computer vision that can notice things that a human, would require a lot of time and attention and expertise to get it done right. And then what the system does, the system was trained on hundreds and thousands of molecularly diagnosed cases, where it learned to recognize them and understand them, and understand when an individual is affected or unaffected, and if affected, then with what, and what are the phenotypes that are present, and even what genes are affected. Then, sorry, just a bit awkward. All right, so the benefit here is that you can identify patients, undiagnosed patients, those who are at the earliest stage of their discovery, of their experience, right from the get-go, and you can cut enormous time from from the diagnostic journey, because for most parents, in many cases, rare disease, they experience it first as a developmental issue. First, they experience it as some form of developmental oddity. This is the first thing that they come with, this is the first thing that they notice, this is the way that they are thinking about that. They notice it before the doctor does. And if you're able to intercept that from the first symptom, you have a chance to capture a lot more patients and a lot earlier in the journey and refer them on the right path a lot sooner, which really gives them a much better chance at a timely therapy and a better outcome. So this is how the tech works. It looks at an image, it knows how to break it down into regions, into parts. It knows how to understand each part individually and in aggregate. It knows how to extract an insight from each of the phenotypes that it sees. And then it essentially works in a form of, for lack of a better term, of consensus between the different areas. Because different areas may suggest different findings, different phenotypes. If there is, say, a consistency in the finding, the system knows that it looks at something that is not just a fluke, but an actual presence or a fact. So what the system does from there, this is how it's being used. This is a picture of a kid. If I removed the blinder, this kid would look absolutely normal. There is nothing outstanding or noteworthy about this child's appearance. But in the interface that you see here, this is Face2Gene. This is our clinician-facing product that a clinician can take the picture, and the system would then see something that by just looking at the kid, you wouldn't notice. And what it would see is that there is a very interesting match. So if you see there are two bars, one on the left, one on the right. These are the color bars that show that, you see this under the label Gestalt, that shows that the system sees a strong match between the picture that you saw here, and it saw a lot of phenotypes that are matching the findings in patients with these syndromes. So it actually says that it sees the phenotypes that are common to conditions like the Williams-Bjorn syndrome or the BLM syndrome or to others. And it shows you how strong the match is, how strong the similarity is. So it doesn't tell you what the condition is, it tells you that's what you can look at. Now, at this point, you can take this guidance, you can consult any form of literature you have, you can dive deeper, but now you have something where you can start from. Now you have something where you can actually, review and go deeper. In addition, the system would also offer you, if you look at this, you have the option to upload your clinical notes or your health record or other symptoms, like maybe the child has seizures, etc. So you add that. And then the AI has a second bar. So next to the bar that says Gestalt, you also have a bar that shows what is the strength of the match of those things that you just manually entered of your clinical notes. So what you see here, I'm just going to use these two examples. One is Williams-Bjorn syndrome, the other is KBG. And what the system would show you then, if you look at KBG, it would show you where it saw the phenotypes. So it's above the eye, maybe about how deep set the eye is, maybe about the ratio between the eyebrow and the eye. It's nuanced things that computer vision can notice, but a human would struggle. However, if you compare this to the match of the Williams-Bjorn syndrome, you will see that the strength is a lot higher. So you can see everything from the curvature of the mouth to the eyes to even the face itself. So the facial geometry is lit up, which really shows that this is probably this condition. So when you look at it as a clinician, you basically now have all the information you need to make a decision. Maybe you should send the child to a test. Maybe you should send them to a geneticist team, etc. So, and this is what it's really built for. This is how the AI actually helps. So, because at this point, you would have, oh, I'm sorry, and you can see this on top. The system even gives you the list of genes that you can work with. So at this point, even if you're a pediatrician, you have the information that you need to generate the referral, to generate the guidance. And I think you have another question.
Yes. Well, this spoke to what you're talking about now, is just diagnostic delays, especially in rare disease, can take years, and it affects outcomes. We all know that to be true. How useful is it for those listening today who are on a long diagnostic journey? What ways would this impact their future?
So I could actually give you the answer as I guess as an example. I think you may have heard the reporter on CNBC on Becky Quick with CNBC Cures. There was a TV segment on it. She's an anchor on TV, and she came out with a story about her daughter having some rare disorder with only 1,700 people with that disorder known in the world. And it's interesting how she tells the story. She tells the story with, hey, at first when we saw this, we thought that our daughter was autistic and maybe with some, I'm sorry, at first we thought it was just some minor developmental issues, and then they became worse, and then more were added, and then we thought maybe she was autistic, and it added up, and then after a while, I don't remember exactly how long, maybe 7 years, maybe or more, it turned out that actually she has a rare disorder. So imagine this child being encountered in a regular developmental review by a pediatrician. A simple picture could have saved those years through the journey that they went through. This all could have been saved. I guess that encapsulates many similar examples. There is another one I would use that we got to know completely by coincidence last year. There was a story from a mother who went on TikTok, and she said that she has a son who was a bit small for his age and kept coming back to her pediatrician, who kept on saying, don't worry about it, he'll grow out of it. And one day I hope I tell the story right. Please forgive me if I'm not getting all the facts right, but the essence is there, where she saw him on stage at some school event, and it turned out that he was so much smaller than all his friends. She said, okay, I can't take it anymore, and she just Googled, of course, and she found our app, and she took a picture. And this was after 7 years in that journey. And long story short, from the moment she Googled and found our app, to the moment she was holding a diagnosis of a rare disease was under 5 weeks. So the 7 years of wait in 5 years to an answer. And she went with that story on TikTok, and it got 2.5 million views in just a few days. And we got thousands and thousands and thousands of people who were flocking to the app, which showed us that this is not a rare case. This is absolutely not a rare case. 2.5 million views and thousands of people who are coming looking for that same answer, because that story or these two examples are the exact examples of how the AI can actually change it. Those 7 years or 9 years are also the difference, not only for the parents. I just want to say something that I did not plan, but I'm part of the effort of kind of let's band together and let's tell the story. But in order to really make a difference, this needs to hit a critical mass for pharma to invest in the research, for payers to support the reimbursement. And the only way you can get to the critical mass is if you use AI tools that would identify a lot of people who are part of this group. They just don't know that they're part of this group. But you would also be able to show to pharma that when they want to run a clinical trial, it's easy to find the patients. Very, very often, a lot of research is not being kicked off because of the difficulty of finding the patients and identifying and doing that and the time it takes and the cost it has. If you can show pharma, if you can show payers that it's solvable, a lot of the things that we're trying to solve would actually be solved because the answer is in the critical mass of these patients.
So I know that a lot of people are probably thinking this. I asked you this when we were talking previously. I cringed a little at sharing a picture. Of course, then as soon as we were off the call, I went and put my daughter's picture in the app. But how does the FDA ensure that its AI is responsible, unbiased, equitable, trustworthy, all while incorporating patient advocacy and lived experience into the tools that you're designing and improving?
It's fascinating to me that so many, and I'm not saying this with any form of criticism whatsoever, I'm really amused by that parents would not hesitate to upload the pictures on Facebook or Instagram or share. Oh, but to check what's actually going, no, there I need to be careful. It's just, my a bit amused observation on this. But specifically, we are a healthcare tool. So we put a lot of effort into this, and we have a patented technology for identifying the pictures and the images. So what the AI really does, it analyzes the soft tissue. It's a classifier. It identifies the embeddings, the relevant embeddings in the image, where the phenotypes are, and what it takes into the engine. It's just those elements that they cannot be reversed engineered into the identity or the appearance of the person. So it's a technology that was developed specifically to address this type of concern. We're able to take a facial picture and submit it to analysis without submitting the picture itself, without submitting the identity. So it's a patented tech, but we're making it available to all parties that want to be that want to be able to do the same. I think it's important to have it in healthcare. But I will address another aspect of it that I'll just show you. This is where the strength is in numbers. So what you see here on the map is the footprint of Face2Gene as it's being used by clinicians worldwide, right? So you see cases everywhere from the US to Europe to Africa to South America to including islands and places I really didn't know existed until they showed up on our map. What it really shows is two things: that it's being trusted by clinicians everywhere, and that the AI gets data from the most diverse group imaginable. So every ethnicity, every race, every form, every shape, they need answers, and the AI learned and learns from this very diverse group. So to make sure that there is no bias, there is no limit, there is no group that would be underserved or unserved. And about the willingness of patients to do that, I would show you this map. This is the Family Health Checker. This is a product that we created specifically for parents and patients and school nurses even. It's available only in the US, where we let the parents upload it, upload the photos, tell a little bit about the health history and symptoms. And the amount of, the number of parents that were starving for this was quite shocking for us. Like, we thought we were going to find like 1,000 families. We have 100,000. And it's insane. Like, people are so much in need to get some guidance so they can be better advocates for their child's health, that, if you're uploading it to Facebook, I think this is a safer place to do that, because this is actually built for healthcare.
Question. Are you finding pathways, so some syndrome, genetic disorders look like each other, and it turns out at the cellular level, the genes are a pathway. Is your data sorting out that way?
So that's such an important question. One of the most difficult things in using phenotypes when you look at a diagnostic decision support is that overlap or similarity between different syndromes. So we built another AI that its job is really to cluster or to aggregate all the similar cohorts that share certain phenotypes. So we actually found exactly to address the point that you raised, right? So that's why our analysis result is never an answer the child or the patient has this condition. The answer is at least 10 options that you should look at with the list of affected genes that you should test for. So we are not the end station. We are the referral to the lab or to the geneticist team. We are those who make that submission to a geneticist team or a lab a lot easier, and really within seconds instead of the many hours that you would otherwise do. So in other words, yes, we work through those who are similar. We find those that make sense to put on that short list. We extract the genes, extract the phenotypes. And I would only say that we don't only do that, because we have all this information ahead of time, what we do, this is in the upcoming version, we generate a letter of medical necessity for the doctor to use. So essentially, we draft it in advance. The doctor can and should edit it, right? But we include in it most of the information that will take a long time to articulate and put together. This is another example of the power of AI. You normally would be able to do this only after you submitted the sample to the lab, and the lab would do this. It's very, because we have all that information ahead of time, we're able to actually do that in the early stage, all in support of helping you to send the case to a lab.
And the samples of different peoples, different, could certainly influence your statistics, right? So in subsets of people from different, regions that have different traits intrinsically from that regional aspect, I'm sure you're going to get variability in the efficacy, right?
So it's not only right, it's even more right than you would think, because the way diseases would manifest themselves across different races, it's different, and the presence and prevalence of this disease in different races may be different. And then the number of people of a certain ethnicity or race are again different. So there are so many variables that but the true answer to that is because of what computer vision can do, the ability to infer and extract meaningful data, even from small samples, is so strong. So this is the power of this particular AI. Everybody knows AI, that it requires a ton of data in order to learn. This AI was created and designed for rare disease, so it's able to extract a lot of information from a relatively small sample. So if you have an ethnicity or a group that has a unique disease expression, there are not many of them, even then the system works. Is it as powerful as somebody who has thousands and no, but it's still very, very, very powerful. Yes.
Go ahead. One more.
I can be loud.
So I'm told.
Yeah. I don't know if this is on. It's probably better. Yeah, you've described this as a system to help enable pediatricians to work directly with labs. At what point, if any, do you think clinical genetics should be involved in this process?
So one of the reasons that we did that is that the message that we heard from clinical genetics is that guys, there are not enough of us. There are a lot of patients. We don't have the time. We just don't have there are not enough people to go ahead and do that, right? So, and the wait times are 9 and 12 and 18 months to get to an appointment at times. One of our goals,
I wish I had put that on my myth list.
yeah. Well, one of the things that we see that could be very, very helpful is that the clinical genetics to actually be most involved when the results come back from the lab. Because if the AI can help you get the patient to the lab, when you have that type of information where clinical genetics can actually apply its incredible knowledge and expertise, it's when the lab test is back. It's when it's done, it's when it's, and then, so this is where ideally a lot of it could be done. This is about helping the parents go and understand what the results are. It's about helping the doctors. It's in the workflow. I would refer to this as in the post-test consultation. If it's helpful, if possible, in the pre-test consultation, if needed. But the more we can help pair clinical genetics with the data that comes from labs and from actual findings, the more impactful they can be. But hey, we're happy to have originally this tool was built for geneticists. We expanded it to pediatricians because of the demand and the need that we saw. So originally this is built for geneticists to sort through and to work through the cases that they have.
We're going to have to end it there. We're going to have a little schedule change here. We're going to have lunch now. In about 10 minutes, Amanda Bergner will give a presentation on neurofibromatosis 1, and then we'll come back and do our panel discussion. Sorry to spring that on you, Corey. But just Amanda has a tight schedule, so she can only come on then. So I want to make sure she has a chance to speak. So go grab your lunch, come on back in, and we'll start up here in about 10 minutes. Okay. Yes, and started while folks fill their plates. I just want to let you know that lunch is being provided by Alexion, and they've arranged the speaker for today, and Andy Augustine has a couple of announcements for you. So Andy, go ahead.
Real quick, I want to thank Dr. Drum and everybody together this Rare Disease Day. It's quite an honor to be here amongst learning all the innovation and development that's going on to support rare disease. I want to thank Dr. Drum. I want to thank.
A little louder.
Oh, I could be louder. Trust me, I can really turn the volume up. But I want to thank everybody for being. Amanda Bergner for taking the time today to have a discussion on understanding NF1 plexiform neurofibromas with a real patient case. Amanda is a professor of genetic counseling at Columbia University Irvine Medical Center. She's the founding director for the Columbia University Program in Genetic Counseling, a genetic counselor in the Department of Neurology, which is pretty relevant for today's discussions. She also serves as a genetic counselor for both the Columbia University Center for Excellence and the Columbia University NF Clinic. Amanda earned her master's degree in genetic counseling from the University of California, Berkeley. Her clinical work is focused on neurogenetics. How about that? She has developed and worked with several multidisciplinary clinics for a variety of neurological and rare diseases. She has extensive experience in the genetic and genomic research with a primary focus on improving care for individuals and families with neurogenetic disorders and increasing access to genetic counseling and genetic testing. Again, thank you, Amanda, for taking time today. Thank you for everybody's in attendance. My last disclaimer is there's a sign-in sheet for your lunch on your table, and if you could be so kind as to clearly print your name, sign your name, and just your title, that would be sufficient for my accounting needs. So without further delay, Amanda.
Thanks, Sandy, and welcome to everyone. Thank you to the meeting organizers for coordinating this lunch and learn. I'm really excited to be with you all for the next 45 minutes or so. We'll be talking about understanding NF1 plexiform neurofibromas with a real patient case. And in my experience, I remember being a genetic counseling student and an early practitioner, and really felt like case-based learning was such an important part of really getting the information down and understanding how this would be relevant for my practice. So I'm hopeful that that is the case for you all today too. This program is sponsored by Alexion, AstraZeneca Rare Disease, and I'm serving as a paid Alexion AstraZeneca Rare Disease speaker today. So we'll start off with an icebreaker challenge. This is something for you to be pondering as you're really getting started with your lunch there, but really thinking about, can you name one of the diagnostic criteria for NF1? And if one comes to you really quickly, maybe you can try to name two. And we're gonna go through them, but something for you to be thinking about as we get underway here. The objectives of our time together today are to really help you recognize NF1 plexiform neurofibroma symptoms and identify signs of PN progression, understand the importance of early referral for treatment, review the value of a multidisciplinary care team in NF1 plexiform neurofibromas, and to discuss a real patient case demonstrating NF1 plexiform neurofibroma diagnosis and management. So we'll start off with a really important fact, which is that NF1 is one of the most common rare genetic disorders and is often detected in early childhood. In my experience, no matter what part of genetic counseling you're practicing or clinical genetics, you're going to come across NF1. It is estimated that about 1 in every 3,000 people worldwide have an NF1 gene alteration. In my clinic here, when I'm talking with people about the frequency of NF1, I will often relate it to if Yankee Stadium was full, this number of people might be diagnosed with NF1. So I actually did a little bit of looking, and I understand y'all's baseball team is the Guardians. And so maybe in Cleveland, if the baseball stadium was full, there might be 12 or 13 people that have NF1 in the stadium. And so that gives you a bit of a sense of the frequency. It's more common than CF and many other rare genetic disorders that you might have heard a little bit more about. NF1 is an autosomal dominant disorder, but really importantly as well, about 50% of cases are spontaneous, meaning that there is not an affected parent when there's an affected child. And so that's a big piece of recognizing NF1 in the clinic, that it's not always going to be something that a parent has been diagnosed with previously. And so taking all of this together based on the prevalence, NF1 can be encountered in primary care a lot. In fact, pediatricians and other primary care providers are really the ones playing a significant role in NF1 diagnosis and early referral. The other thing that we understand really well about NF1 is that it can be a serious disease and the symptomatology can be really diverse. There are over 3,000 known pathogenic variants in NF1, which is one thing that leads to the diversity of symptomatology. But we also see that even people in the same family that would have the same pathogenic variant can have really different symptoms. So this speaks to the important need of personalizing care in the setting of NF1 and really figuring out exactly what's going on for each person that's been diagnosed and is affected, because it could be different even than other people in their same family. So disease manifestation is highly variable, and symptoms can vary in both number and in severity. We know that with NF1 from an early age, people are predisposed to tumor formation. We're going to talk a little bit more about symptoms. Most of the tumors, at least initially, are beginning as benign tumors, and we'll talk some about malignant transformation of some of those tumors. But NF1 results in an increased risk of tumors along the nerves and can really reduce life expectancy. You can see here on the right that for people with NF1, in the light gray shaded bars, the average life expectancy and the mean age at death is around 52 for both people identifying as men and people identifying as women, as compared with the general population of people without NF1, which is considerably higher for both men and women. And so awareness of the latest diagnostic criteria can really help shorten time to diagnosis and get people the treatment that they need. So if you remember our early icebreaker challenge, thinking about one or maybe two of the diagnostic criteria, I have been working in the setting of NF1 for about 25 years now, and the diagnostic criteria have been very much unchanged the entire time. There was an update about 5 years ago in 2021, which added genetic testing to the criteria, but otherwise they have stayed relatively similar because they're so consistently helpful in accurately identifying people who do have NF1. And so we wanted to be sure that you all were well acquainted with some of the most common features that are the diagnostic criteria. So here on the left, you'll see a couple of examples of cafe au lait macules. And so the first piece of diagnostic criteria is having 6 or more of these cafe au lait macules. The size actually matters. So greater than 5 millimeters in people before puberty and greater than 15 millimeters in people after puberty. So I think about that and often talk to people in our clinic about, in childhood, we'd be looking for cafe au lait macules about the size of an eraser on a standard pencil and that diameter. And in after puberty, about the size of a penny, though I understand now pennies have gone out of fashion. So I'll have to think of a different way to describe the size. Post-pubertal. There are 2 different examples of cafe au lait macules on this slide. So the one on the left is what's called a Coast of Maine cafe au lait macule. It's a little bit more rough around the edges, whereas the one immediately to its right is a Coast of California cafe au lait macule, which has a little bit more smooth borders. So the Coast of California cafe au laits are much more common in NF1, but it's important to know that Coast of Maine cafe au laits can also occur. So they're both pictured here. The second diagnostic criteria is axillary or inguinal freckling. In clinic, I often will talk with families about this by saying, freckling in the armpit or in the bathing suit area. We think about this as skin fold freckling or hyperpigmentation in areas that don't have nearly as much or any exposure to the sun on a regular basis. The third criteria is optic pathway glioma. In this image here on the far right, you're seeing bilateral optic pathway glioma. So you can appreciate the sort of, let me see if my pointer is working. These are optic pathway gliomas. So these are tumors that are growing along the optic pathway nerve here on both sides. And so this is bilateral optic pathway glioma. The fourth criteria on the far left is 2 or more iris Lisch nodules. This is, these are what you can think of as almost like hyperpigmentation in the iris of the eyes or 2 or more choroidal abnormalities. The next criteria is 2 or more neurofibromas of any type. So here pictured are cutaneous neurofibromas, which are growing on the surface of the skin. And immediately to the right of the image of cutaneous neurofibromas is a plexiform neurofibroma here in the neck and the shoulder region. So the criteria are for 2 or more neurofibromas of any type or a single or more plexiform tumor. So right away, we know that plexiform neurofibromas are really indicative of NF1. Immediately to the right is the next criteria, which is having distinctive osseous or bony lesions. What's pictured here is sphenoid dysplasia or sphenoid wing dysplasia. This is the same person with 3 years of time elapsing between the 2 photographs. And so you can see that there's a difference in the bone formation, and there's actually a missing bone here in the sphenoid wing, which can change the shape of the bones of the face. And then the update that was made in 2021 was to add a criteria of having a heterozygous pathogenic NF1 variant with a variant allele fraction approximating 50% in apparently normal tissue, such as white blood cells. So on a blood, buccal, or saliva test, if an NF1 pathogenic variant is found with variant allele fraction approximating 50%, that in and of itself is its own criteria. And so for an individual having 2 or more of these criteria, or if they have 1 and they have a parent who themselves has 2 or more of these criteria, that leads to a diagnosis of NF1. So having familiarity with each of these criteria are really helpful in identifying people early on that have NF1 and being able to get them to appropriate services. We know that NF1 plexiform neurofibromas specifically are fairly prevalent. Up to about half of all people with NF1 will have at least one plexiform tumor, and that they can be progressive and debilitating. These start off as benign nerve sheath tumors that occur early. The hypothesis is that people are born with plexiform neurofibromas. They may not become visible or large enough to be noticed until they're later on in life, but that they are always present from birth and can progress over time. In fact, a fairly sizable study that included more than 250 people showed that over 3/4 of people in the study had plexiform neurofibromas that were either invasive to the tissue around or displacing, meaning that they were interfering with the local site and what should be happening anatomically there. Importantly, for plexiform neurofibromas, while they initially begin as benign nerve sheath tumors, there's an 8 to 13% lifetime risk that a plexiform would transform into a malignant peripheral nerve sheath tumor. MPNSTs are aggressive and life-threatening metastasizing tumors, and they are a high cause of morbidity and mortality in the setting of NF1. So really important that plexiform neurofibromas are followed carefully and closely over time. We know that plexiform neurofibromas can emerge anywhere on the body, so this slide gives us a couple of examples. This is definitely not all of the places that PNs can form, but on the far left is an example of a plexiform neurofibroma on the back, so sort of towards the upper and right part of this image. In the middle, an example of a plexiform neurofibroma of the cervical spine, and you can see that it was initiating in the cervical spine, but has invaded into the neck and shoulder region as well. And then an example of a plexiform neurofibroma on the arm here on the far right. One of the things that clinical practice has taught me is outlined here too, which is that many internal plexiform neurofibromas can only be seen on MRI. And so a physical exam may not reveal all the plexiform neurofibromas that a person may have. And advanced imaging plays an essential role in diagnosis and in disease management for plexiform tumors specifically. We know that plexiform neurofibromas can progress rapidly, and results from the NF1 natural history study out of the National Cancer Institute over the last few years took a look at progression of plexiform neurofibromas over about a 5.5-year period. And the vast majority of people that had plexiform neurofibromas in the setting of NF1 had tumor progression. So plexiforms are often measured based on the volume of the tumor by volumetric MRI. And so we saw from this study that the median plexiform neurofibroma growth rate was approximately 16% change in volume over 5.5 years. And that in general, plexiform neurofibromas were growing continuously for people enrolled in the study. So one thing that we may think is, well, if this is happening in childhood and we see a growth of plexiform neurofibromas volume as someone is getting older through childhood, maybe that's just part of their whole body growing as part of childhood. But what we actually see is that the volume growth rates for plexiform neurofibromas can exceed weight or height growth rates. So that really suggests that an increase in the size of the PN is not solely related to normal childhood growth, but is actually beyond that. So in the setting of NF1 and plexiform neurofibromas, early action is really crucial. So we know that PNs can grow fastest during the first 10 years of life, and they do typically continue to grow beyond that. And that the morbidities related to plexiform neurofibromas can worsen over time, and that they're highly variable and can potentially cause life-altering complications. And so we wanted to share a few things with you related to some of the key takeaways that pediatricians and other primary care providers, I would say, one of the things we're seeing a lot in our clinical practice is that people that are even working outside of these spaces and maybe outside of neurology spaces too, such as cancer genetic counselors, are often finding NF1 is coming up on genetic test results or broad scale cancer panels, things like this. And so I think it really underscores the fact that any of us, no matter what setting we're working in, will likely come across NF1. And the more familiar we are with the manifestations and the clinical criteria, the quicker we're going to recognize, identify people, and refer them to the appropriate specialists. And so I think probably every one of us plays a critical role in really improving outcomes for people, especially in the setting of plexiform neurofibromas, which we need to keep a close eye on with early referral to an NF1 specialist, or if there is not an NF1 specialist right nearby, to an oncologist. And so just starting to think about in your own practice, and maybe some of you all are in training, and so thinking some about how you can take a role in ensuring that NF1 and or NF1 plexiform neurofibroma symptoms aren't missed, particularly given that maybe some of them are going to be internal and not visible on a physical exam. So in thinking some about the lived experience of people with plexiform neurofibromas, I think this can be really helpful to get a sense of what is the actual impact for people. And so probably one of the biggest is physical morbidities. So plexiform neurofibromas can be quite deforming because they can be invasive and displacing. And so there can be visible deformities throughout the body, which I imagine you all have appreciated just from the couple of images that we've seen already. People can experience vision impairment, airway compromise. Plexiform neurofibroma-related pain throughout the body is an important symptom. We're going to talk a little more about that. Certainly motor dysfunction, depending on where the tumor is growing and what it might be affecting around it. And bladder and bowel dysfunction, these are some of the most intrusive symptoms into people's daily lives that affect activities of daily living. We also know that plexiform neurofibromas can cause cognitive impairment, impairing attention and executive function, as well as contributing to learning difficulties. And there can be significant psychosocial impacts, including anxiety, depression, certainly stigma and psychological stress, and difficulties with peer relationships as well. We'll talk a little more about this too. And we know that morbidities for plexiform neurofibromas can occur and worsen as tumor volume increases, which is yet another reason that early detection and referral to the right type of specialist can be really important for people living with NF1 and plexiform neurofibromas. So as mentioned a moment ago, pain is a substantial morbidity for people with NF1 plexiform tumors. And in a study of people with large, which in this study was defined as 3 centimeters or greater, which again, I'm big on having a really lived experience of this. So that's about the width of your thumb. If you take a quick look at one of your thumbs or a neighbor's thumb, that's about 3 centimeters. In this study, they were looking at plexiform neurofibromas that size or larger, and pain was the most reported morbidity. Pain can be chronic, it can be episodic, or it can be both. It can be localized to where the plexiform neurofibroma is, or it can actually be experienced as diffuse pain that's occurring away from that location and can interfere regularly with daily activities. And, you know, in studies of pain, there can be lots of different things that are measured, but one of the really consistent things is use of pain medication. And in one study looking at NF1 and plexiform neurofibroma, it found that the faster growing plexiforms were associated with increased use of pain medication. So it does help us recognize that pain really is a substantial part of living with a plexiform neurofibroma. And as mentioned a few moments ago, the burden of living with plexiform neurofibroma can go beyond the physical symptoms. There is a higher incidence of psychosocial challenges for people living with plexiform neurofibroma. So children and adolescents have a higher likelihood of developing an anxiety disorder. And one of the primary concerns that parents report about children living with plexiform neurofibromas was sadness. And so if we take a look at, in this particular study, about 30% of parents were reporting that their children were always or often sad. And adults report recalling childhood feelings of relationship failure, school failure, failure at work, and low self-esteem. And I think our case study may help shed a little light on exactly what's contributing. But if we think about some of the physical morbidities and changes in activities of daily living, in addition to some of the emotional sequelae that can really impact people's daily life. Patients should be monitored for signs of plexiform progression, and so expert consensus recommendations are that patients with NF1 initially need to be assessed for plexiform neurofibroma through careful examination, and then even after the initial assessment, monitored for plexiform growth, even if there wasn't a plexiform initially located, depending on how the first exam was done. Regional MRI and whole body MRI are also really helpful, and these are the imaging modalities of choice. It's helpful to work closely with an NF1 specialist here to try to limit the number of MRIs and the frequency of MRIs to just what's needed in the setting of NF1 and plexiform neurofibromas. Patients with known plexiforms do undergo MRI surveillance at particular intervals appropriate for them, depending on the age of the patient, the location of the tumor, the presence of morbidities related to plexiform tumors, how the plexiforms look on imaging, and the history of prior plexiform growth. And so patients also are monitored for onset or change in plexiform-related pain. And in my experience, that's a lot of the education that's happening in clinic for patients, for patients' families, for parents, how to recognize when there are changes in plexiform tumors. And that's an important thing for them to be partners on, so that they're circling back if there are any changes that are noticed. Just taking a moment to talk about surgery for NF1 plexiforms, there are a lot of challenges to surgery. Not all plexiforms require intervention, but when surgery is feasible, it can be an effective way of doing what is called debulking, or reducing the bulk or the volume of a plexiform tumor through surgical resection. But not all plexiforms are operable, and not all are fully resectable, because they do continue to grow within and throughout the nerve sheath. They're often proximal to vital organs, making surgery difficult. Plexiform tumors notoriously have poorly defined margins, which make them very hard to achieve total resection or completely remove, and are often in challenging locations such as the head, neck, spine, or trunk. We've already seen a few images of that in the presentation today. And we know that plexiforms can be hypervascularized, which can increase the risk of bleeding during surgery. And so you can see here on the right that there are noticeable and important percentages of postoperative complications when using surgery in the setting of plexiform neurofibromas. And even if surgery can be done and can be done smoothly, we actually see that plexiform regrowth is very common. So looking at rates of plexiform neurofibroma regrowth after surgery, they range from anywhere from 20 to almost 70% likelihood that it will grow back. And it seems dependent on the extent of resection. So on the left here, we can see that even completely excised or full resection tumors have about a 20% chance of recurring. Near total resections, where greater than 90% of the tumor bulk was removed, have a 40%, so double, 40% chance of regrowing. And subtotal resections and partial resections, the numbers go up from there. So even with successful surgery, plexiform neurofibroma regrowth is common. So we know in summary that plexiform neurofibromas can lead to serious clinical complications. They impact people across the lifespan and across multiple domains of their lives. Vigilance is needed. Detection at an early age is really helpful because we know morbidities can worsen over time, and getting people into specialized care sooner than later is really helpful, not just for managing plexiform neurofibromas, but all of the morbidities that can be associated. And that complete surgical removal is not always possible, and that regrowth is common. So maybe you're starting to think about how you ensure that plexiform neurofibromas are treated before symptoms deteriorate, especially during early childhood. We know that shared decision-making with patients, families, and providers really lead to positive patient outcomes far more often. So many studies have shown that patients and parents want to participate in treatment decisions, and that this desire has been increasing over time. Maybe this is something you all have noticed in your own practices to date. And caregivers in the setting of daily life and the way that plexiform tumors can impact people's daily lives can provide really crucial insight into people's condition and their children's condition or relatives' condition in terms of the impact of these morbidities on things related to pain, sleep, anxiety, depression. And a systematic review actually has shown that better outcomes are likely when there's shared decision-making happening. There's improved satisfaction and people have less decisional conflict. So this is a really important part of working with the person, with the family that's living with plexiform neurofibromas and NF1. So in addition to the affected patient, NF1 can actually impact parents and unaffected siblings as well. We see in a study of caregivers of children and adolescents with NF1 plexiforms in the US that about half reported anxiety, and we can compare this to the general population frequency of about 20%, so more than double. And about 35% reported depression, again, compared to the general population of about 20%. Unaffected siblings of children with rare disease such as NF1 also suffer from more psychosocial problems than their counterparts. And caregivers are reporting a substantial impact on work and daily activities by way of mean productivity loss and regular activities that are impaired on average as being a caregiver. So we know that the impact of this is not just on the person living with NF1 and plexiform tumors, but also on the entire family. Caregivers have an important role not only in communicating how things are going and helping to add to the understanding of the impact of plexiform tumors on someone's life, but also, as we talked about before, in helping to notice changes and possible progression. In chronic conditions, shared decision-making is preferred by a lot of patients and families. And we know that for affected patients, there definitely is an impact, but we see that there's an impact on other families in the setting of NF1 as well. And so really starting to think about what the practice has been in your clinic for engaging caregivers. Are there any things you would maybe want to adjust? That's a question we asked ourselves regularly in our clinic here. And we know that a multidisciplinary team can strengthen patients' care. This is probably true across the board, but is particularly true in a multi-system condition like NF1. And so here is a list of some, but definitely not all medical specialties that may be part of a multidisciplinary care team. I would certainly add genetic counselors and many other providers that are a really important part of working together with a family to strengthen patient care. And so beyond immediate medical support, additional services that are available for patients and families, and actually in my experience are particularly helpful in the day-to-day life of living with NF1 and plexiform tumors, are nurse coordinators and child life specialists. Psychologists certainly can help build strategies for coping with psychological impact, including anxiety and depression, which we saw can be really increased, both for people living with NF1 and their family members. And advocacy groups play a huge role, so including the Children's Tumor Foundation, the Neurofibromatosis Network, and we'll talk a little bit more about the NF Collective here in a moment. But bringing together a community that shares the same life journey can provide a lot of support for people. I'll just share from my own experience that I've been attending NF summer camp for the last probably decade and a half, and there's 80 or 90 people living with NF1 that spend the week at summer camp, and it's such a huge piece of promoting resilience for these people, these young children and teenagers that are living with NF1, and being able to learn about their own condition, being able to build community together and become advocates for themselves. So maybe starting to think about how to connect patients that you're seeing with NF1 advocacy groups, referring them to specialized NF1 centers, and all of these can be really beneficial. We do know that consideration and careful consideration is important to be successful in the transition of patients from pediatric to adult care. This is probably true across the board, but particularly in the setting of NF1, it's a complex multi-system disorder, and it does require long-term monitoring and psychosocial care. So you can recall a few slides ago, we talked about the NCI study that showed the rate of plexiform neurofibroma growth just in a 5.5-year period. And so if we think about that transition from adolescence into young adulthood, very often in our clinic, one of our experiences is that people might go away to college after high school and be at college for 4 years, sometimes, a little less, a little more. And just in that time, if they sort of, take a break from their NF clinic, that's about 4.5 years of time. And we saw the rate that plexiform neurofibromas can grow. And so just an important thing to keep in mind that this time of transition is a bit of a vulnerable period. And so being very thoughtful about that. We want to be sure that you're aware of the NF1 track, which is an NF1 specific tool that can actually be used in your clinic to assess transition readiness. It's a questionnaire that you can use with your adolescent or young adult patients that have NF1 to help assess their transition readiness from pediatric to adult care to take on coordination of their own care. And multidisciplinary collaboration is really helpful to facilitate an effective transition here. To be sure that people are not, lost to follow-up in this time period where they're needing to change potentially a lot of their care providers. As mentioned, there are a number of advocacy groups, physicians, and centers that specialize in NF1 across the U.S. Want to make sure you all are aware of nfcollective.org. This is the NF Collective. It's a group of organizations that are all dedicated to improving the lives of people living with NF and their families by providing accurate and reliable information and NF resources. So that's a good place to check out. You can also access a physician locator there and have a sense of where centers are that specialize in NF1 across the U.S. So multidisciplinary care is really important in the setting of NF1. It's needed to address the symptoms and the challenges that people have with NF1. Prompt referral to an NF1 specialist and an NF1 center, or if there isn't a local center, an oncologist can be really helpful, again, to reduce those longer term morbidities for people living with NF1 and plexiform tumors. And so, again, this comes back to all of us being aware of the signs, the symptoms, and how to identify people that might have plexiform neurofibromas in the setting of NF1. So we wanted to wrap up today with a case study, again, being able to apply this to all of this learning that we've done to an actual case and have a sense of how this might look from a clinical perspective. So we'll introduce you to Joseph, who was 2 years old at diagnosis. Joseph has a parent with NF1, but I see this all the time in the clinic. This is a parent that didn't know they had NF1 and was diagnosed after their child was diagnosed. So this is very common that a child will come to medical attention, receive a diagnosis of NF1, and then there's that piece of, 50-50, is it de novo in the child or was it inherited? And we do a close exam in our clinic of both parents if they're available. And oftentimes there is an affected parent that has very mild features and never came to medical attention, but does have NF1. And so that's the case for Joseph. And Joseph's journey began in early childhood with his pediatrician. So at about 18 months of age, his mom took him for a well child visit and asked about several café au lait macules she had noticed in the axillary region and on the lower back. And these were monitored by the pediatrician, and by about age 2, his pediatrician also detected a small lump on his left ankle foot, and it continued to increase in size over time. So Joseph's presentation with café au lait macules and a growth on his foot raised suspicion for NF1. His pediatrician referred him to a neurologist, who then referred him to a geneticist and an oncologist. The geneticist confirmed the presence of an NF1 gene alteration, so through genetic testing. And the oncologist and neurologist had consults together to determine that the foot growth was a plexiform neurofibroma. And so at age 2, Joseph was diagnosed with NF1 and with a plexiform neurofibroma. And so his management included years of active surveillance in young childhood and repeat surgeries. So from age 2, when he was first diagnosed and a plexiform was noticed, through age 11, He received several debulking surgeries. So these are going to be subtotal resections of the neurofibroma, that plexiform tumor in the foot or ankle, and was followed originally by a neurologist who was monitoring the neurofibroma and managing his pain with medication. And so this is a fairly common thing in our experience in terms of ongoing management of plexiform tumors. So between ages 4 and 11, Joseph also saw an orthopedic surgeon and had several surgeries for insertion and removal of pins to help stabilize the foot and alleviate some of the symptoms that Joseph was experiencing. He had several more surgeries between ages 9 and 11 and worked a lot with physical therapy and experienced chronic pain throughout this time, which again, we know is a really common experience around plexiform tumors. His walking was also compromised, and as is often the case in our experience in our clinic here, people with foot-ankle plexiform tumors, if they continue to grow, have a really difficult time being able to use a shoe on that foot. And so at this point in time, Joseph couldn't wear a pair of shoes or wear a shoe on that foot. And at age 9, Joseph showed signs of withdrawal and depression, being unable to participate in normal activities with other children his age. So again, this is really common and something we see a lot in our clinic here. And it's important to be tracking all of this simultaneously with the medical complications of plexiforms. So his management included support for his caregivers too, as well as Joseph, his parents needed emotional support as they navigated through the process of feeling overwhelmed and alone while managing their child's care, not having had experience with this, which is really common in our clinic here too. And initially, the family thought that there were limited options for Joseph, but by being connected with an NF1 center, which is a multidisciplinary approach, which we've already seen can take a look at a lot of the different morbidities related to plexiform neurofibromas, Joseph was able to receive care from a multidisciplinary team that could address a lot of his different needs, his family's needs. And over time, a comprehensive treatment plan was established. So a nurse coordinator, child life specialist, and psychologist provided additional support and resources that the family needed on top of medical management. And today, in Joseph's case, his plexiform neurofibroma and symptoms are being managed. There are probably many positive outcomes to this case, but one of the ones that can make the biggest impact, especially for children in early to middle childhood, is highlighted here, which is that he can now wear shoes, and a shoe particularly on the foot with the plexiform, which can make a huge difference in day-to-day life and being able to participate in activities. So just to wrap things up for today, pediatricians, other primary care providers, and all of us, no matter where we're working, really can play a critical role in early detection of NF1 symptoms and facilitating early referral to NF1 specialists or oncologists. Early action can be crucial, especially in the setting of plexiform neurofibromas that typically grow fastest in the first 10 years of life and the morbidities that can worsen over time. As with cancer and other chronic illnesses, engagement of both patients and caregivers can enhance the care of people with NF1 and plexiform tumors. And there's a network of specialized NF1 centers that can provide comprehensive multidisciplinary care, as well as ancillary patient services. So keeping in mind the updated diagnostic criteria, they're all pictured here on the slide, and we've been through these, that clinical diagnosis of NF1 can be made if someone has 2 or more of these signs, or 1 or more of these signs and a parent who has 2 or more of these signs. And so with that, I'll leave you with a question that from my experience as an educator here in our genetic counseling graduate program, I often ask our students, come Monday morning, what's going to be different after this talk? So what are you taking away? What are you taking away from this talk, and what information have we discussed that's going to affect your approach to NF1, or sort of being on the lookout for NF1 and being ready to refer people? So I do want to say that your feedback is really important. There are always ongoing updates and improvements being made. So would ask if you have a phone nearby, you pull it out and use the QR code to complete the post-program survey. My understanding is you all have a schedule, as do I have a schedule. I'm happy to provide answers to any questions offline, and I know that Andy's there to provide answers to questions as well. I really appreciate the invitation to be with you all. I hope your lunch was delicious and that you have a good rest of your conference. And greetings from snowy, snowy New York City. Thank you very much.
Thank you, Amanda, and thank you, everybody, for being here today, and thank you, Dr. Drum and the Planning Committee for allowing myself and my colleagues here, along with Alexion. We have the privilege to have our patient advocacy, patient education team here. A lot of the topics that Amanda discussed, specifically like the diagnostic criteria and other things. I have literature here. Please feel free to stop by, take whatever you like, and if you wanted to do the post-program survey, I can print out the copy if you didn't get a copy of the QR code. So thank you all. I hope, and like Amanda said, I hope you enjoyed your lunch as well. Thank you.
So one more session, and this is a chance for a little more interaction with the audience as well. Corey, would you mind bringing up and Tiffany and Ann, I think were our, if I'm not forgetting. So this is our panel discussion, and again, to get some more insight. And while they come up, I also want to thank again our sponsors. It's been a great event. One of the researchers who has left just a few minutes ago just told me, he said he thought this was spectacular to be able to see the interaction between, or all the facets of rare diseases, right? It's people affected, the advocates, the basic scientists, the clinicians. So it's a very broad group. All right, Corey, you're in charge.
I hope everyone can stay awake for a little while after lunch. Thanks for sticking with us. So this panel is really about navigating life services and support with rare disease. And I think after talking with these wonderful women, there is so much going on in between all the appointments that happen when you have a child with a rare disease. And that's what navigating life really is. And so we're going to talk about the steep learning curve from being thrown into a diagnosis and how to navigate that and how to find the support that you need, because there is a huge need for community and resources, and they've all become experts on finding them. So we're going to talk a little bit about that. So if you guys will each introduce yourselves, tell a little bit about your story, and then we'll talk about some resources.
Well, I think that everybody has met me by now, but I'm Tiffany Sammons. I'm a community engagement manager at NORD, and I have an 8-year-old with Coffin-Siris syndrome and newly Klippel-Feil syndrome as well. So happy to be with you all.
I'm Ann Clark, and my son Jude has 15q24 microdeletion syndrome. We don't have a name yet, but I am his full-time caregiver. I used to teach, but after diagnosis, he just had a lot of medical complications and pretty severe developmental delays. So I am full-time coordinator.
Hi, Pam Judge here. I'm the mother of Connor. I'm different from them from the standpoint that my son was stricken with a rare disease when he was an adult. He was 22. He was diagnosed with NMO, but we now know that it's MOGAD. And the rest is history.
We made Pam join the panel, and we're glad to have you join us. I'm also a caregiver to my daughter who was diagnosed when she was 15 years old. So it's things have changed a lot over the last 10 years, and we've learned a lot, and really sharing that is so important. I know Anne brings so much wisdom from caring for her son for so long, and I just want to ask, like, where did you find the resources first, and what was the most helpful for your family?
I could go on, so yeah. I'll keep it in 3 sections. The first one is the medical. So when he was first diagnosed, I walked around with my binder. I had all the symptoms, which doctor, when was our last appointment, the blood work, all the information. And around 2021, Jude's OT, his therapist, who unfortunately couldn't join us today, she told me about the complex care unit that had opened up at the Cleveland Clinic. And she had been up there and met the doctors, so she knew that Jude would be a candidate for that. So once he was a part of the team, they were able to coordinate blood work and help me with surgeries. Whenever he's in crisis or emergency, they're able to coordinate with the specialist because they're all on our building on the same floor, for those who are familiar. And it just took a lot of the time of coordinating away from me and more of the caretaking that I needed to do. And overall, it's just, again, all of these things, the quality of life for him and our family really changed everything. The second piece is the education. So when he was first born, the pediatrician connected us with Help Me Grow. And he did that. And then right around 3, he had a multi-factored assessment and got on IEP. Because of my teaching experience, I was able to put the autism diagnosis for the scholarship fund through the state. So those things helped. And kind of at the same time, we moved to Chagrin, where we had another assessment, and they used the least restrictive environment and an outside placement. So he was able to go to schooling at KidsLink, which is in Twinsburg, and then he is now at Proactive in Chesterland, and their focus is adaptive skills, communication, safety awareness, and that has been great because a lot of times they want to throw letters and numbers out in front of him, and that's not where he's going to be successful. And now he is doing pre-vocational skills at 13. He works for a half hour stacking the shelves at Drug Mart. So he's very happy in his life and feels a great sense of purpose. So again, just improving that quality of life. And the last part is the county waiver. So originally we started off with the family support program, which gave you $750 a year, which you can use for a variety of different things, camps and respite care. As he grew and the span between his actual age and his developmental age became bigger, his OT again suggested the county waiver, and it is a Medicaid waiver. It's level 1, and the big portion of that with the Medicaid is that they look at just Jude's income, not the household. So with this waiver, he gets Medicaid. So not only do we have our TRICARE, then he also has Medicaid, and it has other benefits. All of his diapers and wipes and pads are covered. We also get up to 20 hours a week of provider support, which is so life-changing for our family. It's my night. I get to go be mom to my 6 and 7-year-old. I know that Jude is getting out swimming. He's using his communication device out in the community. So it's just, they're all life-changing, and they really increase our quality of life. But I think the big lesson learned is it's all about the people who tell you about it. So that's why today is so important.
Right, and tenacity, because that process is not an easy one to apply for those programs and have denials, and you have to have tenacity, and you have to keep picking yourself up because it's an exhausting process.
Yeah.
And go ahead.
Do you want me to?
Yeah, a little.
So the waiver has been such an amazing, life-changing thing, like I said. But as part of the process to get on the waiver, initially Jude's SA said we will have a phone interview. So I had the phone interview, and afterwards I got a denial letter. And I appealed. They said you didn't submit any letters of eligibility. News to me. So I did go out and get those letters of eligibility from doctors and therapists and teachers, submitted it. denial letter again, and I asked for the assessment. And when I looked at the assessment is where I found inaccuracies. So they had checked the box for him that said he does not elope. When in 2020, I used the lifetime maximum that the county does give to put a fence around our house because he was eloping. So I did have to correct that. And also in the summary from our phone conversation, they had written that he receives infusions for safety awareness. So I'm sure that everyone can understand that he receives infusions for his immune deficiency. So I appealed. I appealed and went to the state, and the state ended up overturning the county's decision, and we were able to get on it. And moving forward, there's always a lot of paperwork. There's this ISP plan, there's the monthly check-ins, searching for a provider, going through the interviews, all of that. So it definitely takes a lot of persistence and a lot of you doing your own homework, but it is worth it when you have it all set up.
That's a lot. So Tiffany, do you want to talk about, I think you can speak to this because you have worked through NORD so much. There's a lot of things that we don't know we need as rare parents until we're kind of thrown into it, and we need that support system around us to help kind of guide, like, you are going to need this or you might need this. Like, how does NORD help with that? And how have you been, like, your experience with that yourself?
There was so much that I just didn't know existed until I needed it, until another parent that had gone before me shared that resource with me. I didn't know about county funds. I didn't know that his diapers, pull-ups, wipes could be covered. I didn't know about respite programs or respite providers. And I worked clinically for years. So that goes to show there's a lot to learn, and it's a learning curve. NORD has so much to offer on their website that it's a bit daunting for families that are newly diagnosed at times, but I would encourage you to look at the patient resources. We have financial assistance. We have respite program I spoke about this morning. I was even asked earlier today on a break about, do we help internationally? We are a national organization. However, we have sister organizations across the globe that we can point you to. We work a lot with Eurordis. We have partners in India. We have partners in Canada. And so everyone at NORD will go out of their way to try to find you a resource if we can't provide it ourselves. And that's what we're here for. So I think it's very overwhelming. When you first get a diagnosis, it's like you can't even put a foot in front of the other, yet alone think about how you're going to handle a year from now or 5 years from now. And so let those who have gone before you show you the ropes. Don't be afraid to ask for help. As somebody that never asks for help, I'm telling you, burnout's a real thing. So don't be afraid to lean on your community. I think events like this are so impactful because you find your community. I often say at NORD events, welcome to my family reunion, because that's what it feels like in the rare disease space. And so that, I guess, would be my biggest plug.
Pam, do you want to talk about that a little? The impact of finding community and for you even creating your own? I mean,
well, finding community, way back in 2014 when my son got diagnosed specifically with NMO, the community was on the West Coast. Nothing else existed except the former Patient Health Alliance, Guthrie Jackson. And our family will be indebted to them forever and ever because they at least gave us hope. There was no treatment. There was nothing. Like, basically, we learned back then, a third of the patients get better, a third of the patients are a little messed up, and a third of the patients are very messed up. Putting that in, my mom's vocabulary, right? Without that support, I don't think our family would have. I mean, we were very blessed to find doctors at the Cleveland Clinic who to this day are on my speed dial. I mean, what doctors do that, except doctors that are above and beyond. So we need to do a shout out to your doctor and our doctor, Dr. Mary Rensel. Yeah, just incredible. And then with our, the next thing that happened is then because my son was paralyzed and had to learn how to walk again, then he went to Metro for 6 weeks. And again, we had incredible doctors at Metro, so we can't say enough good things. So we had a good experience, but I think a lot of it had to do because I had my daughter in the family who understands all the medical jargon and who could help navigate what treatments Connor should be on, et cetera, et cetera. So I think in each of our stories, there's something that like moves you along, but you cannot just sit there. You have to like take the next step. You have to ask questions. You have to self-advocate every step of the way.
Absolutely, self-advocate and advocate for your family if it's a child. And not take no for an answer in a lot of ways. We learn to be very careful with our words and not have a, meltdown, but there are times where we have to push to get the things we need for our kids, whether it's like appealing a denial or just, continuing to come back because we know something is the matter. As far as finding the resources that you use for the state, is there anything else that you want to share about like school resources or anything? I think that we also talked about the resources that Jenny was going to share that she's helped connect you with that are like beyond just medication and diapers. Like there's some like other things that our kids deserve to be part of too.
Yeah, so I would say that Jenny, this person I keep talking about, she, so a little background about her, she actually had a sister with cerebral palsy, and then she became an occupational therapist, and she was involved in a lot of different things. Jude started with her at 10 months old, and she just, she becomes your friend and your family and who I was texting with at night. And she, all throughout his 13 years, she looked at what his need was at that time, and she was able to identify, or if she wasn't sure, she knew who to contact to find out different things. So there was a short time where he needed a walker while he was learning how to walk, and they knew that it wouldn't be permanent and like not to have to go through all the insurance and all of that. So they told me about Matthews Lending Library. And you can go and get equipment there and borrow it for a period of time. Then there was Young Athletes, which you guys might be aware of. And it was a place for him to go, even though he did not understand how to play different sports and do all that. He was able to go and be with other peers. And so there was just different things all throughout. She was the one that told me about the support, family supports and the waiver. She actually told me about the maximum lifetime spend through the county as well. So when we needed the fence, she also told me about the complex care team as well. So it was a lot of just like, this is what's happening right now. This is what I need. And having her help me with that. I'm trying to think of.
Oh, and the rec.
Oh, yeah. You're right. Two more things. So he has a feeding tube, and we finally got to the point where we didn't want to sit still. And he also gets infusions for 2 hours every other week. So she helped us through the seating and mobility clinic at the Cleveland Clinic. And we have gotten a stroller, we have gotten a feeding chair, and also a 5-point harness in the car because he started surprising me climbing up in the front while I was driving down the highway. So now we have the 5-point harness so he can't get out, which is really great. And now that he has the provider, at our home, I wanted to get him moving more and doing different things. He loves swimming, but other ideas. And she told me about Rec to Connect, which is a Cleveland organization, and there's East Side, West Side locations, and they're all different types of activities. So there's like rock climbing, there's drumming, so there's all different activities, and you can pick a different activity and actually use the waiver funds to pay for some of them.
I bet that's new news for some people here. I've learned a lot just getting to know you through this experience. I have to step way back because when my daughter was diagnosed, I didn't even know what a 504 or an IEP were. I had 4 healthy kids until I all of a sudden was in the rare disease world. Like nobody explained any of that. And Pam and I always say, like, we should have a book that we get when we get a diagnosis. Like, who's going to tell us? And then Tiffany said, well, I got a book. So
there was no manual. It took a few years, but my son went to an ongoing therapy program called the Perlman Center. He went there twice a week where he received multiple therapies, PT, OT, speech, feeding, and had a teacher. And they became my best friends too. Like, you don't get out much when you're a rare mom in the beginning, so you get really close with these people. But when he started there, they handed me a binder, and apparently nobody else gets this. And it had some really awesome resources. It had accessible playgrounds. It had information of how to get a hold of the Board of DD. It had places with allergy-friendly food options. It had basic things, not so much your medical things. But then in the back, it had steps to filling out the waiver through Medicaid and just really tangible things. And at the time I was like, man, this is really daunting. But over time, just looking back through, that was really helpful. NORD also has a state-based resource library. So we try to have people from each state add to that over time, so you can find all sorts of things that might be applicable. If you are a parent or caregiver, this is where you take out your pen and write all these resources that took us years to find. But May We Help was really big in our life for the same reasons. Insurance typically will only cover one mobility device in a 7 to 10 year time frame. So if you need a medical stroller and you need a gait trainer and you need a bath chair, you better choose wisely. And so May We Help has an equipment swap every quarter. And if you don't have something to swap, they will still service you free of charge. And so we were able to trade in his gait trainers when he used one. That way we could save the insurance in case he needed a medical stroller. Now my little redhead decided to defy a lot of odds, and we didn't need all of the things that we thought we would. But it's helpful to be able to have resources like that. So May We Help was a huge one. I'm trying to think. A Kid Again. So there's an event or a program here in Ohio called A Kid Again. Any child with a rare chronic disease qualifies, and they do events every other month, I believe, to try to get kids to feel like a kid again, that are accessible and friendly. And there's something for everyone. Like my kid hates crowds. If he was in this room right now, he would be running out the back door. So they have all sorts of options that really are good for a variety of ages and a variety of skills.
In NORD's programs as well, there's so many programs.
Yeah, so I'd be remiss to say if I didn't mention this. Living Rare, Living Stronger is a program that we offer patients and families. It used to be a national program, and we decided that was a greater need than one city once a year. And so this year we are bringing that program to 3 locations and 1 Spanish-speaking virtually. And so we're trying to meet more people where they're at. And I have the honor of getting to lead that program. So it's free for all patients and caregivers. And we talk about a lot of these things, even in more depth. And it's awesome. I went to one, obviously, last year, and there was a mom that stood up and said, my kid has that same rare disease, and I've never met anybody else. And so it's kind of an arm hair raising moment, but but it's really neat to be in a room of people that just get it.
It's a profound sense of belonging. I would encourage anyone that finds themselves in this situation to find community. I mean, it might be a very small Facebook group, and maybe you'll not meet them for 3 years, but get connected. It is so valuable, and find people like these ladies. It will help.
The Facebook group page that I have for the 15Q24 for Jude is where I've actually gotten information and actually taken it to the doctors, and then they've tested him for it, and they're like, yes, he has an immune deficiency. But it was talking to those people on that Facebook group page. And I actually attended, the whole family did, Rare Disease Day out in California. Is that October of 2024? And we got to meet one other person with 15Q, because there's only about 100, 150 worldwide. So that was, I thought I was looking at my future, because he was 30. So it was, and they just sat right next to each other and looked at each other. They're both nonverbal. So it was a pretty amazing moment too. Yeah.
I would also just encourage everybody to kind of open their eyes. I encounter so many patients that come to me and they said, do you think there'll be somebody there that has the same disease like that story you just shared? There are so many rare diseases. I mean, I think we've redefined as over 10,000 now. And so the chances of being in a room of 300 and meeting somebody with your exact disease is pretty slim. But we have different disease states amongst us, and we have so many similar stories and challenges. So I think the rare community has a lot to offer everybody in the rare space, because you're experiencing a lot of the same things. Isolation, insurance barriers to, you name it. So don't think that you have to make this small community even smaller.
To close, is there anything that you would say to a family that's at the beginning of their journey, that's listening virtually, or someone in the room?
I would say you're not alone. Make sure that you keep your persistence, even if it's hard to navigate and find those resources. You are running a marathon, not a sprint, and you definitely need to take the time to rest and recharge when you can too.
I'd say worry about today, not tomorrow, not 5 years from now. I caught myself up in that a lot in the beginning. And the other thing I would just say is anybody that's new to this journey, that's sat up here and listened to all of us speak about where we're at now, know that we didn't start here. Don't be overwhelmed thinking you have to go create a new therapeutic or you have to open a nonprofit. You can make a lot of impact in a lot of ways, and it takes time to get there. So be okay with where you're at now and know that we didn't start there either. I think there's like 5 phases of grief, and you go through them and back again, and every day is different. And so don't feel like you're not doing enough just because you're listening to people that have lived it for 8 years, 13 years, 10 years. So
I agree with everything that they said. And because my son's a little older, again, what a difference a day makes, because back in 2014, there was no treatment, and now we have a number of different drugs. One that I am proud to say that my son was the first person in the entire world to be on the clinical trial. So there's hope. Just always have hope. And whoever you meet in the rare disease space, especially if you've met me, then link in with me, and we will be friends for life, and we will do good things together, because these are all the people I work with, and I'm very blessed.
Thank you for the hard work to pull this together for the last 7 years.
Yes, thank you, Pam.
Yeah. No one that's in the rare disease space as a parent or a patient, like chose that. Like these diseases are not caused by something we did, even though we all have a lot of guilt, because that's where moms, that's where, that's still something we're all accepting, I'm sure. I know I'm still doing that. But for all the clinicians in here and students, you're choosing to help our families, and thank you. Thank you for the work you're doing. It is incredibly impactful, and we see you. Thanks for listening.
Closing remarks. If you're parked here below the building, we still, I think, have 6 QR codes to get you out of there for free. So I hope you take away so many things from today. I think Tracy's title kind of summed it up, ripple effect. It's how everybody affects everybody else. Part of my ripple effect, the people who check you in, my wife and my daughter, so they have been dragged along through a lot of the things that we do. They also, Julie, Catherine, where are you, Julie? But Julie brought zebras, so please take one along with you, remind you of what we're doing here today. And one more time, thank you to all of our sponsors. And Pam, there just aren't words to say. Thank you for what you've done. So.
Every single person in this room, again, the sponsors, the clinicians, legislature, the caregivers, the patients, Case, and the incredible Dr. Drum, who it happens to be his birthday, so let's give him a hand. Again, I hope you know how much you're appreciated, and we look forward to all the good work we're going to do together. And to keep the momentum going, I just wanted to do a shout out to Katie Harris of Harrington Discovery Institute, because she got them to host our 8th Rare Disease Day next year.